everybody. If you are in the audience, can someone please give me a thumbs up to make sure that you can hear me? All right. Thank you. All right, welcome everybody to our 8th annual Women in Data Science at Lane Medical Library conference. We will start with a short welcome video from the main organization, and then we'll get started.
Has been around since 2015. These products are small, as only organizations delegate cannabis, and the brand that is distinguished for their outstanding demonstrating work in data science. And everyone, they're all over the
So technology happens. We tested the sound about 3 times before the conference started, but apparently audience cannot hear the sound. I apologize for that. You will receive a recording of the program as well as the slide deck, and you'll be able to watch. It's only a 2-minute long video, but I apologize for technical difficulties that we're starting with. It happens. Anyhow, let's regroup. And I hope everybody is excited to be here today, and I would like to welcome you to our 8th annual Women in Data Science and Lane Medical Library conference. My name is Boglárka Hadoston, and I am the manager of research and instruction here at Lane Medical Library. Here is our land acknowledgement. I will give you a second to take a look at it, as we would like to recognize the land that we stand on. And I would also like to use this as a gentle reminder that we do have a code of conduct, and thankfully I have never had to refer people to it, so I would like to continue with that tradition itself. But if you do witness or encounter any kind of behavior that does not follow our code of conduct, please let me know, and I will attend to it. All right, very quickly, here is our agenda and a couple of housekeeping items. We have a Q&A box at the bottom of your screen. So if you ask questions of our presenters, please use the Q&A box. If you do see a question that you would like answered faster or you would like to upvote, then use the like button right next to the question and it will move it up in the queue. Live captioning is available for the duration of the conference. You have to turn it on your own computer and your own screen. Just click the little CC button at the bottom of your screen to view the captions. Just our agenda very quickly. We will begin with an opening keynote, or an opening remarks, I'm sorry. We will begin with opening remarks by Dr. Colleen Cuddy, who is the Director of Research and Academic Collaboration here at Lane Medical Library. Then Dr. Marina Sirota will deliver the opening keynote. After her, we will have a panel discussion. We'll transition over to it and we will meet our three panelists. I will be moderating the discussion. We do have some pre-set questions, but we will also be taking questions from the audience. And again, please put your questions into the Q&A box. After this, we'll take a 5-minute break for everybody to stretch and regroup. And then at 11:10, we'll have Dr. Johnson deliver the closing keynote. The conference is being recorded, as I just mentioned, and if you need to step away at any time, you will receive the recording via email once the event concludes, probably within 48 hours. And before I turn it over to Dr. Kadi, I would like to thank the people without whom this event would not exist. Their dedication and hard work in the past 6-ish or so months ensures that this event is a success and that we can continue in the future. So Colleen, Amanda, Connie, Danielle, and Yuping, you are all greatly appreciated, more than you know, and I can never tell you with words. So at this time, I will turn it over to Dr. Colleen Kadi, who is our Director of Research and Academic Collaboration here at Lane Medical Library. And she will welcome us and open up this event. Colleen, take it away.
Thank you, Bogi. Good morning, good afternoon, and good evening to everyone joining us today. My name is Colleen Kadi. As Bogi said, I'm the Director of Research and Academic Collaboration at Lane Library. And on behalf of Stanford University, it's my great pleasure to welcome you to our 8th annual conference, Women in Data Science Satellite Conference. And you'll hear us say that a lot today. We're very proud that we've been doing this for 8 years. And it's really evolved from a small in-person local gathering held in the physical library to this global virtual forum. And that's really a testament to the power of our connected community and the importance of this platform. It's particularly inspiring to see such a strong international network of women in the audience who are driving innovation and leading the conversation. It's not just our distinguished speakers, but so many of you who are in the audience. Your collective expertise is really what makes this field so dynamic. I think it's especially fitting that we are gathering this year during Love Data Week to explore the theme of Ready, Fair, Go, making clinical and biomedical data findable, accessible, interoperable, and reusable. We need an acronym for that as well, I think, Bogi. I encourage you to really check out some of the other events happening at Stanford and around the world for Love Data Week as well. As a representative of Lane Library, this theme is central to our mission. Libraries are essential partners in the research lifecycle, providing the expertise and infrastructure to help researchers manage, curate, and share their data effectively, ensuring that it is truly fair from its inception and it remains a valuable asset for the global research community. This work is more critical than ever. Applying FAIR principles to sensitive clinical data introduces profound challenges, and we'll probably hear quite a bit about that today. We need to really carefully navigate these complex ethical considerations and maintain privacy protections for our patients, balancing this drive for open science with our fundamental responsibility to protect patient trust. With new regulations like the NIH data management and sharing policy, these principles have moved really from this idea and this ideal to a core requirement. So this makes a discussion such as the one we're having today really essential. Our goal today is to provide you with both inspiration and some practical strategies to navigate this landscape. As Bogi already mentioned, we have an exceptional program to guide the discussion. We begin with a keynote from Dr. Marina Sirota, followed by a panel moderated by Bogi with Dr. Teresa Zayas-Caban, Dr. Ashley Griffin, and Dr. Daniela Meeker. And we'll conclude with a closing keynote from Dr. Erica Johnson. Again, I just want to thank you for being here to share your time and insights. I, again, want to echo Bogi's thanks for our Lane Library staff for pulling this all together and everyone who's here. And I'm delighted to turn it over to Bogi, who will kick off the program.
All right, sorry about that, so many buttons. Thank you, Colleen, for that lovely welcome. And with that, I am honored to introduce our opening keynote speaker, Dr. Marina Sirota. Marina is currently a professor and the interim director at the Bakar Computational Health Sciences Institute at UCSF. Prior to that, she has worked as a senior research scientist at Pfizer, where she focused on developing precision medicine strategies in drug discovery. She completed her PhD in biomedical informatics at Stanford University under the mentorship of Dr. Atul Bute. Dr. Sirota's research experience in translational bioinformatics spans nearly 20 years, during which she has co-authored over 170 scientific publications. Her research interests lie in developing computational integrative methods and applying these approaches in the context of disease diagnostics and therapeutics with a special focus on women's health. The Sirota Laboratory is funded by NIA NLM, NIAMS, Pfizer, March of Dimes, and the Burroughs Wellcome Fund. As a young leader in the field, she has been awarded the AMIA Young Investigator Award in 2017. She leads the UCSF March of Dimes Prematurity Research Center at UCSF, as well as co-directs INACT, a center to study precision medicine for endometriosis. Dr. Sirota also is the founding director of the AI for All program at UCSF, with the goal of introducing high school girls to applications of AI and machine learning in biomedicine. She is here today to give us the opening keynote. Marina, welcome. Thank you for joining us. And I will stop sharing so you can share your screen.
Sounds good. Thank you so much for inviting me to present and share some of our work, and also for the kind introduction. As Bogi mentioned, I trained at Stanford, and I trained both undergraduate and graduate school at Stanford, so it's kind of like coming home for me. Can you see my regular screen or the presenter view? Good one. Okay. All right, so I'd like to tell you today about some of our efforts integrating different types of data for discovery, and we use both molecular and clinical data in our work. I'll talk about ways that we use data that's publicly available, some of the data sharing efforts that we lead within my team, and also what kind of science that can lead to. And hopefully that'll open up some ideas for conversation and discussion afterwards. So again, I'm Marina, I'm from UCSF, and I always like to start by saying why I'm excited to be a computational biologist today, why I'm excited to be at the intersection of biology, medicine, and computer science. And one of those reasons is there's a lot of data that's available out there. Over the past 20 years, the technologies that we use to profile the human body, both in health and disease, has advanced tremendously. We've been able to use genomics and transcriptomics to look at health and disease, but we can also look at antibody repertoire, epigenetics, proteomics, metabolomics, the microbiome, and now many of these measures on a single cell level. Also, we have access to real-world data. This is clinical data that gets captured when somebody goes to see a doctor. Structured data includes diagnosis codes, lab test results, demographics, and other information. We have also access to de-identified clinical notes. And this provides a real-world patient data set to explore health and disease, even though these data are incredibly messy and have not been collected for research purposes to begin with. And finally, we have computational technologies. We've been doing statistical analysis and traditional AI for a very long time, but now we have opportunities to integrate these approaches with generative AI methods. And really, this allows us to bring all of these data sources together. So it's an exciting time. In my lab, we focus on ways to figure out and bring different data sets together, both molecular and clinical data, with a focus on women's health. So we study pregnancy outcomes like preterm birth and recurrent pregnancy loss. We study autoimmune diseases, which tend to occur more often in women than men. And we have a pretty large research program in endometriosis. Sorry, in Alzheimer's disease and neurodegeneration. We also work on endometriosis, but I will not be talking about that today. So today I'll focus on our work relating to pregnancy outcomes and specifically preterm birth.
I'm sorry, Marina.
Yes.
You have a little pop-up screen on the top of your
Oh,
presentation.
okay. I'm not sure how that happened.
It
Okay,
went away. Yeah, there you go.
is that better? Okay, thanks for
It's much
letting me know.
better.
Yeah,
Thank you.
I don't quite know how, oops, it's jumping ahead of me. Okay. All right, let's see if,
It looks good.
okay. Oops, no more, huh? Okay.
Now we can see you.
I know, slight technical difficulties. Give me a second. So something happened to my presentation, but I will revive it.
All right.
Okay. 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 Are we recovered?
We're
Not yet.
seeing your the presenter mode. Sorry.
Okay. How about
There
that?
you go.
Okay. All right. We're
Perfect.
back. Okay. So preterm birth is defined as live birth before week 37 of gestation, and worldwide, about 15 million babies are born premature. 1 million of these infants die within the first 28 days of life. And in nearly half the cases, we don't really know why the woman goes into labor early. So it's a complicated phenotype. This is a figure out of a review paper from Roberto Romero that demonstrates the various things that can go wrong and can impact parturition timing. And that includes vascular disorders of the placenta, infection, uterine overdistension, cervical disease, so maybe the opening of the cervix, the breakdown of maternal fetal tolerance, what's going on between the immune system of the mom and the baby, as well as stress. And of course, many of the factors we still don't know. It's a heterogeneous problem and it requires a heterogeneous data-driven solution. And in my lab over the years, we've been using different types of data to better understand pregnancy parturition and why babies are born early. I won't have time to talk about all of this today, but I'll touch upon a few of the examples. We started out by exploring genetics of preterm birth. We know that moms who are born preterm are much more likely to deliver preterm themselves, that this type of phenotype tends to co-occur in siblings. So there's a known genetic component. And we started exploring this. This was actually when I was a research scientist at Stanford, as part of the Prematurity Research Center at Stanford at the time, in collaboration with David Stevenson and his team. And we saw that actually when we looked at genetics and we looked at baby genetics, we couldn't find too many significant signals. It's unclear whether it's because the population of patients that we were looking at was very heterogeneous. Maybe we didn't have the right sample size. But it was my sort of first foray into starting to understand preterm birth. And there was more questions than answers. We started exploring environmental exposure signals, and this is in collaboration with Tracy Woodruff at UCSF. And we started seeing things like water contaminants, arsenic, and nitrate being associated with adverse pregnancy outcomes. Then we started looking at gene expression, microbiome, and finally electronic medical records. And I'll touch upon those in a little bit more detail in this presentation. So in terms of gene expression analysis, this gene expression is whether certain genes are turned on or turned off. And we really wanted to identify some relevant biomarkers. Previous studies have been limited by sample size and focused on placental samples. We really wanted to focus on maternal samples here. This work was led by a very talented graduate student in my lab, Bianca Vora. This was actually her rotation project. And she started out by aggregating data from the public domain. So these are publicly available data sets from a gene expression omnibus repository. And we queried them for data that contained maternal blood signals and had outcomes, preterm birth versus not. At that time, there were only 3 studies available, and these are microarray studies, so the technology is quite dated. Nonetheless, we had an opportunity to bring these data sets together. So these graphs are dimensionality reduction plots. I believe you can see my cursor, right? Okay. And here, this plot shows that before normalization, when we visualize these data sets, they cluster out by data set. But we can overcome that heterogeneity by bringing all the data together. Here they're colored by study, and here they're colored by case control. And you can see a slight shift, but the signature, it doesn't separate beautifully. This is just showing that the timing of sampling was around the same time. So then we apply some statistical analysis and we look for genes that are significantly differentially expressed. And this is a heat map visualizing those results. The samples are on the x-axis and genes are on the y-axis. This is showing 210 genes that are significantly different in moms who end up delivering preterm. And this purple bar, in light purple, are women who deliver preterm, and in dark purple are women who deliver at term. And even here, you don't see a clear separation. We see that there's a cluster here in the middle that is enriched for, or potentially enriched for women who deliver preterm, but it's not black and white. We can start studying the biology of these genes, though. We can look at networks, how they're connected. These are the downregulated genes. These are the upregulated genes. And we can look at the pathways. And the pathways, we saw a lot of immune dysregulation. We saw that the innate immune system was upregulated in women who deliver preterm, and the adaptive immune system was downregulated. We can also look at specific markers. So these are specific genes and proteins that are secreted, and the ones that are highlighted in purple are also ones that appear in an earlier time point. I didn't draw attention to this, but one of the data sets had an earlier time point of sampling, T2 second trimester versus T3 third trimester. So these were interesting. We wanted to dig into those a little bit further. IL-1 receptor 1 is a cytokine receptor that belongs to the interleukin family. It's been associated with a number of autoimmune diseases and investigated in the context of choroamnitis. IL-1 receptors 1 and 2 are proteins that that have been found in abundance in rats, in the rat uterus actually, and regulated by progesterone, a hormone that's really important in parturition. TFPI is tissue factor pathway inhibitor that is involved in blood coagulation. The product of factor 10 is involved in early onset preeclampsia and associated with placental development. So basically, this was telling us that the pathways that we're identifying, the specific genes that could act as biomarkers, are biologically relevant and interesting to explore. And this was only possible by bringing these data sets, these disparate data sets together to get additional power. So this initial study was published in 2018. We did a number of follow-up studies exploring this inverse relationship between maternal signals and fetal signals, looking at immune repertoire. But really, we were interested in taking what we found here and seeing if we can apply it to drug discovery. One of the approaches that I've devoted my career to studying for my graduate studies at Stanford is drug repurposing, or identifying new uses for drugs that are already FDA approved. The motivation behind drug repurposing is pretty simple. It's very expensive and it takes a long time to take a drug and bring it to market, and 90% of drugs fail in early development. A solution to that is to explore drugs that are already FDA approved and see if we can identify new uses for them, which both reduces the cost of development as well as the risk of failure. So we developed a pipeline for computational drug repurposing. Given a gene expression signature of a disease, such as the one we just saw for preterm birth, we can query that against a database of drug gene expression data, and for each drug, identify whether the gene expression signature of the disease is reversed by that drug or not. And this is not fancy AI. This is plain statistics. We're looking for something like an inverse correlation. We use a non-parametric rank-based approach called the Kolmogorov-Smirnov test, but we're looking for that negative relationship. And the hypothesis is if the disease signature is reversed by a certain drug, then maybe that drug could be a therapeutic. So the original paper came out now almost 15 years ago. We've applied it to a number of different disease areas, including IBD, dermatomyositis. We've explored a number of cancer applications. The drug expression is actually measured on cancer cell lines. So that makes a lot of sense. We can match it very, very well. During COVID, of course, we tried this as a drug repurposing approach for COVID-19. But today I'd like to focus on our work relating to preterm birth. And this was led by Brian Lee, a very talented postdoc in my lab, in very close collaboration with David Stevenson, who runs the Prematurity Research Center at Stanford. So again, we wanted to take this methodology that I just mentioned and apply it to identify new therapeutics for preterm birth. We took that signature that I just talked about and we queried it against the data set that I mentioned. It's called the connectivity map. It was developed by the Broad Institute, and they took about 1,300 drugs. They put them on cancer cell lines and measured gene expression, genome-wide gene expression, before and after. There's a second data set called LINKS, where they looked at a lot more drugs, over 12,000 drugs, but only measured 1,000 genes. And in our hands, the imputation of the rest of the genome or transcriptome for that set of, I don't know what's happening. Can you guys still see? It popped up again, huh? Okay, hopefully this is all right. So it wasn't as good. So we focused on the connectivity map data set. We also, because we're dealing with women who are pregnant, a vulnerable population, we narrowed down the drugs to pregnancy categories A and B for this type of analysis. So this is what the results look like. We identified 83 drugs that significantly reversed that preterm birth signature, and 13 of them were part of pregnancy, FDA pregnancy AB categories. Out of 13, 4 have been previously investigated for preterm birth. So this is what the data looks like. On this heat map, you see the preterm birth signature that I mentioned before, and these are the drugs that reverse it. You see that progesterone is at the kind of end of this list. Progesterone at the time was the only FDA-approved drug to prevent preterm birth. It doesn't work great. It works about 1/3 of the time, but nonetheless, it was good to see something that has been tried before. The other 3 compounds that have been previously explored include metformin, folic acid, and clotrimazole. On the left-hand side, you can see the network of these drugs. So the drugs are these kind of pinkish rectangles, and the genes or their therapeutic targets are the ovals. And you can see when we look at progesterone, there's another drug called lansoprazole that's very close to it in the network connectivity. We saw that it shares some therapeutic targets, and we thought that it might be worth investigating. So turns out lansoprazole is a proton pump inhibitor. It's available over the counter. It can induce a stress response protein called heme oxygenase 1. And David and his team has seen before, they were very interested in studying this protein. Their whole lab is devoted to studying heme oxygenase 1. But they've seen before that it has been shown to induce pregnancy loss in HO deficient mice. So our hypothesis was that if we're targeting the expression of this gene, then maybe lansoprazole could be used in preventing recurrent pregnancy loss in preterm birth. So we partnered with David and his team
Sorry about interrupting. We see the
Yes.
presenter mode again.
Okay.
Sorry.
I'm not sure. Okay, is that better?
There you go. Yes.
Okay.
Thank you.
No problem. Thanks for letting me know.
First.
Okay, so we embarked on this mouse model study where it's an LPS-induced inflammation-based fetal wastage model. And this is what the timeline of the experiment looks like. The pregnancy is confirmed. The animals are treated with LPS, and we included 2 groups, treatment with progesterone, which is the standard of care, and treatment with lansoprazole around the same time. And then we look at the outcome as the number of viable fetuses. And this is what the data looks like. This is just the animal model. These are normal animals, as well as 3 different versions of the model, LPS 100, LPS plus oil, and LPS plus DMSO. And those are the vehicles used to deliver the various drugs. And this is looking after treatment. So you can see that treatment with lansoprazole recovers the phenotype, while treatment with progesterone does not. This is a small number of animals. Pregnancy in animals is very different than pregnancy in women, But nonetheless, this gives you an idea of ways that you can use public data and relatively simple statistics to come up with a biological hypothesis that's potentially worth testing. So then we started collaborating with Dr. Sing Wei in Cincinnati, who has a different animal model. This is an immune-based animal model of pregnancy. And they found the depletion of T regulatory cells causes fetal wastage and the loss of live pups. They've shown that this expansion of these cells are also specific to this specific phenotype. And their paper came out a few years ago. They've also shown that treatment with progesterone alleviates this phenotype. So here you can see percent of fetal wastage goes down. 17-OHPC is a version of progesterone, and the number of live pups goes up. And this is showing those specific cells that get depleted with treatment. So Sing and his team, repeated this experiment in their model with lansoprazole, and these are the results. This is the phenotype of that's controls. This is where the model in red is induced, and blue and green are treatment with lansoprazole and famotidine, which is another drug that achieves a similar clinical effect to lansoprazole. And you can see significant reduction with lansoprazole treatment in fetal wastage. The number of pups goes up significantly. And then these are the specific immune cells that we've been interested in. So this is incredibly exciting. Now we've been able to validate this finding in 2 independent animal models. And we're currently working with Brice Goodnier at Stanford and his team, who've developed a multiplexed mass tag barcoding drug screen to look at compounds in human samples. So this is given a blood sample from women who deliver at term and women who deliver preterm, and they're able to put a number of compounds, including lansoprazole, on these human samples. Through that, this is a very complicated slide. It's still a work in progress, but through that, we're able to see how these drugs are actually working and which immune cells they're modulating and how. So hopefully I've showed you with the gene expression data analysis that we can use publicly available data to understand some of the biology that's going on in terms of potential biomarkers and pathways. We can further use it to identify therapeutic candidates. We work very, very closely with wet lab collaborators. My lab is entirely a dry lab, but we work with wet lab collaborators to validate these findings. So we worked with David Stevenson and Sing Wei. And then we're currently working with Brice and his team to understand what these drugs might be doing immunologically in human samples. We're also expanding this approach to other relevant conditions such as preeclampsia and endometriosis. So I want to step back. I told you about how we can use data that's already publicly available. And this really drove us to expand and develop a data repository for preterm birth research. We were asked by the March of Dimes to launch a data repository. This was started out by myself and the late Dr. Atul Butte, and now co-led by myself and Dr. Timiko Oskotsky in my group. So we've created this database called pretermbirthdb.org. Please check it out if you're interested. It has a lot of data. It's meant to serve as a central data repository for molecular data across the 7 March of Dimes transdisciplinary centers. It includes transcriptomics, genomics, microbiome, proteomics, and the whole idea is to enable new scientific questions, enhance collaboration and coordination, both among the March of Dimes centers and beyond, and accelerate the pace of discovery. This is the growth of the repository. It was launched in 2017, and then in 2020, it became its own kind of website and resource. We also published a paper about it now a number of years ago, but everything still holds. The data repository has grown. It has 75 studies across over 30,000 participants and 45,000 experimental samples. We also track the downloads and whether people are using the data. And I believe we're past the 10,000 download mark by now. This slide is a few years old. This is what the data looks like. You can search by study, assay type, biosample, which center it comes from, as well as condition. The majority is preterm birth, but there's some preeclampsia and other relevant pregnancy-related phenotypes. So once we started creating this repository, we kind of looked and saw, well, what other types of data are available? And we noticed that there's a lot of microbiome data out there that's been generated for pregnancy. We started out with Dr. Edith Costi, who was a very talented postdoc in my lab at the time, and launched a meta-analysis journey and learned that microbiome data is messy and complicated and hard to put together. So we started with a few independent studies. That's what was available to us at the time. And when we aggregated them, we saw that women who delivered preterm had a more diverse vaginal microbiome, especially early in pregnancy, which we did not see in women who delivered at term. And this, I mean, it makes sense. The gut microbiome, you want it to be diverse. The vaginal microbiome, not so much. And we could identify specific bacteria that are significantly associated with preterm birth, especially in the first trimester. But again, we learned that these data are complicated, they're hard to put together. But we realized that there's a lot more to do. So we partnered with a group called DREAM, and I'm currently also one of the DREAM directors. So if you're interested in doing this type of work, please reach out. We launched a crowdsourcing challenge. The DREAM challenges are meant to bring the computational scientific community to a certain biomedical question. And the goal here was predicting preterm birth from vaginal microbiome data. It really truly takes a village. So this paper came out 2 years ago in Cell Reports Medicine, and it was spearheaded by Jonathan Golub and Tomiko Oskotsky, but many, many groups across different teams collaborated on this effort. I think we had over 50 authors on this paper. This is what the data look like. We started with 9 publicly available data sets. The ones that are in purple were part of our repository. The ones that are in white, we were bringing into our repository from under other sources. And you can see that they vary in the number of participants. This is all sequencing of the 16S vaginal microbiome gene, but different primers are used for the sequencing. There are varying number of samples, but all in all, it's a huge data set. So we had data across 3,500 over 3,500 samples that we started with, 2,500 that were term and about 1,000 that were preterm, including 328 that was early preterm. We normalized and aggregated all of this data with an approach called Maliampi. I'll touch upon it in a minute, and it was developed by one of our close collaborators, Jonathan Golub. And this is the timeline of this challenge. We launched it in June of 2022, and it took a few months. Then we had teams participating from all over the world. We had 300 teams who registered. You can see the map here. Over 100 submissions for each sub-challenge. And then we evaluated the models and created ensemble models using the approaches that were brought forth by the community. This is what the data look like. Again, a large number of samples, very, very exciting. And there were 2 sub-challenges, predicting preterm birth or predicting of early preterm birth. Why do we want to combine data across studies? We want to make sure that our results are generalizable and that we're able to validate what we find in different ways. But as I said, microbiome data is complex. So Jonathan developed an approach called Maliampi, and it's available in the public domain, so please use it, that allows us to take all the sequences from all these studies and build a huge phylogenetic tree. And once that's done, we can get all the studies on the same ground. So this is what the data looks like. Each dot here is a sample, and they're colored by study. This is before normalization. You can see they, just like that early transcriptomics data, they cluster out by study. But when we get all of them together, it looks much better. The ones and X's are our validation data sets. So as part of this effort, we generated 2 independent validation data sets to test the models. But when we color by case control, you can appreciate that really it's hard to distinguish which women deliver at term and which women deliver preterm. And this is where the power of machine learning comes in. So the 2 validation data sets, one was generated with Dr. Adi Tarka and Roberto Romero at Wayne State University, and the second one was done in collaboration with Stanford, David Stevenson, and Nima Agapur and their respective teams. Again, all the data was harmonized and released to the community. These were our results. The top teams performed at about 0.7 accuracy for prediction of preterm birth and 0.87 for prediction of early preterm birth. It's not too surprising. Early preterm birth is a much stronger phenotype, so it's easier to predict. These are the winning teams. They all got to come out and present their work in Las Vegas at the Dream Challenge conference. And it was a great experience to bring these international teams together. There were a few teams from the US, there was a team from Italy, a team from Korea. We had 2 winners per sub-challenge. And then we can start understanding the biology. What are the features that are predictive? And we saw some things that we knew about, like Lactobacillus crispatus, Gardnerella vaginalis, but then we can identify some new interesting associations too. And you can see that they're pretty consistent across the models. We can also bring the models together and build ensemble models that tend to perform better. So for sub-challenge 1, we could get to 0.74, and for sub-challenge 2, up to 0.91. And this was work done by Alice Tang, a very talented MD PhD student in the lab. So now the data, the paper is available. Please take a look. The method for data processing is available. We also created a visualization tool for those who are not computationally inclined called VMAP. It allows you to explore the data through a user interface. That's available as well. And the presentations of the teams are on YouTube. This data set we're very excited to share became one of NSF selected NAIR resources. And the idea of this whole NAIR kind of effort is to enable infrastructure to drive AI innovation and discovery. This is one of 10 data sets. It's the only one that deals with health and disease and actually specific to women's health. Other ones have to do with shipwrecks and cell painting and other types of data documents. So we were very excited that this data set was recognized by the research community as a significant resource for health AI development. So stepping back, crowdsourcing is very promising and exciting. But in the last several years, generative AI has come to everybody's attention as a tool for leveraging and understanding data better. So about a year ago, we launched a project, and it was led by a master's level researcher and a high school student in my lab in collaboration with Dr. Adi Tarka, to take 3 of these dream challenges relating to reproductive health, including the microbiome one that I mentioned, and ask AI models, AI large language models, to build models, predictive models on this data. So we gathered 3 of these, and this is the description of the study. The study is coming out next week in a journal called Cell Reports Medicine. And we evaluated 8 different large language models given a prompt. And I don't expect you to read the prompt in details, but the idea was to compare and see how AI would perform in a competition like this. And honestly, we were blown away by 2 things. One is across the three groups, you can see the human top performers are in blue, and the generative AI models are in the other colors. And you can see for question 1, for Dream Challenge 1 and Dream Challenge 2, they do very comparably across both AI and the human teams. In red, you see the average performance. I want to point out that RMSE, we want it to be lower. For AUC, we want it to be higher. Here, you can appreciate that the human teams, the winning teams do better, but the AI does on par with the average performance of the human teams. But the most incredible part of this study is it only took a few months. Each one of the dream challenges took several months for the teams to build the models, for us to evaluate them, and then a significantly longer time for us to write the paper. This whole project was carried out in less than 6 months across 3 different questions. So the potential of using generative AI to speed up the scientific process is incredible. Of course, we have to be careful, but there is an incredible opportunity here. So hopefully I've convinced you that using publicly available data and enabling data sharing can help move science forward. I want to end with an example of leveraging electronic medical record data for research. And this started out as also a high school student-driven project in my lab by Carolyn Wang under the mentorship of Edith Costi. She was at that time a rising senior at Gunn High School in Palo Alto, and she ended up going to a conference and presenting this work, which I was very proud of. So we wanted to ask whether we can use electronic medical records to study preterm birth. And the reason this was our first foray into clinical data analysis, and the reason we thought it was a good idea was, well, the women deliver in the hospital, so we have their outcome information. Pregnancy is a 9-month time frame, so we can align the trajectories. And well, this is arguable. There's some consistency of care, like first trimester test, second trimester test, so forth. So at that time, we had data on about 15,000 pregnancies. We have demographic, lab test results, diagnoses, medications. And we started building these trajectories, starting kind of backwards with the date of delivery, seeing what information do we have during pregnancy and what information do we have on these women prior to pregnancy. The data is messy, very messy. We start out with about 15,000 pregnancies. By the time we clean the data, according to timelines, outcomes, and so forth, we cut the data set in half. But when we started looking at associations, again, this is simple statistics, not yet getting into predictive modeling, we see some things that we knew about before. For instance, preterm birth, women who are Black or African American are at a higher risk for preterm birth, and we can see that using demographics. When we look at risk factors, prior preterm birth is the biggest risk factor for delivering preterm. Other factors are obesity, type 2 diabetes, hypertension. This is not too surprising. But when we look down the list, we saw things like general anxiety disorder, major depressive disorder, hyperthyroidism. This is still quite interesting. When we were doing this work, I ended up presenting this at a collaborative meeting and learned that a group at Vanderbilt that led by Tony Capra was thinking along the same lines. So instead of competing with each other, we joined forces. And this work came out in BMC Medicine in 2022. They were starting to do the same thing with the goal of predictive modeling. There was a cohort identified at UCSF that I was just talking about, as well as a cohort at Vanderbilt. And the initial predictive models were built at Vanderbilt. They sent them over, and we were able to run them at UCSF. And this is quite remarkable. The performance is all right. We get about 0.7, 0.8 accuracy from the clinical records, but you can see that it's consistent across Vanderbilt and UCSF, which is very encouraging. The patient populations are different, the physicians are different, but the models held up. This was our first foray into this type of analysis. We've since collaborated a lot with various groups, including Nima Agapur at Stanford. This work came out, a joint publication came out in 2023 in Science Translational Medicine, where we were interested not only in predicting maternal outcomes using EHR data, but predicting neonatal outcomes from maternal records. Here we expanded the data sets. Again, the models were built at Stanford and then sent over to our team and verified at UCSF. And the first time we ran these models, they didn't work. And we were starting to dig into why. So this is what we saw. We saw that there were certain clinical codes that were used at Stanford that were not used at UCSF at all. So if you look, this is plotting of the clinical ICD codes at Stanford versus UCSF. There's a number of codes that are on this x-axis. There is 0 at UCSF. And they were part of the model. So they were kind of messing things up for us. Once we restricted to the codes that are used across the two institutions, we showed that for 5 different phenotypes, we're able to validate the clinical models from Stanford in a completely independent cohort of patients at UCSF, which is quite remarkable. And this is using maternal clinical records to predict what happens to the babies. So this is the data that we have now. We have data on over 30,000 births at UCSF, 25,000 moms over the past 12 years. We know whether they deliver preterm or a term, and within preterm, we could look at indicated versus spontaneous. So we've done some phenotyping work to explore that. We also have data across the UC system. That's nearly 250,000 births. And you can see the demographics of those populations. These are incredible resources. Stepping back, we have data on 8 million patients at UCSF, including over 40 years of data. We have de-identified clinical notes. We have imaging information. We have limited genomics sequencing, focusing on a cancer panel. And these were all efforts spearheaded by the late Dr. Atul Butte when he came to UCSF to start the Baker Computational Health Sciences Institute. Across the UC system, we have broader data. So 6 campuses, only 12 years of data, 9 million patients, and about 40,000 cancer genomes. So you can appreciate that we have an inherent discovery and validation system here. We're able to leverage large language models on this data. We have HIPAA-compliant implementations of various models that we can explore using Versa, an internal system. And there are a number of groups on campus who are working very hard to democratize these resources to the broader community. So we have about 1,000 computational users of the Information Commons platform, but Rohit Vashisht is a research scientist at the Baker Institute who's developing a system to interact with clinical notes using a kind of chatbot interface called Dive EHR, and then building foundation models using this data. And then Sergio Baranzini has developed MedCP, which focuses on structured data integration and analysis. So hopefully I've convinced you that it's an exciting time and there's an exciting opportunity to take data, whether we're dealing with molecular data such as DNA, RNA, cellular models, tissue models, to better understand disease using physiological models, and bringing in population level analysis such as electronic medical records. There's a number of faculty, around the world, but also at UCSF as part of Baker, who are working across the spectrum, bringing these things together. So with that, I'd like to thank my team on the left-hand side, everybody leading the various projects I had a chance to talk about, our collaborators at UCSF, both in the context of preterm birth, but also computational. The Peraton team develops the data repository that I mentioned. As you saw, I collaborate very closely with many, many folks at Stanford. So again, it's very exciting to be here presenting to this group. The DREAM group, it's been an amazing effort, and we all learn a lot from each other, as well as funding by the March of Dimes and support of the March of Dimes network. And this is my team at UCSF, and I'm happy to answer any questions.
Thank you so much, Marina. That was incredibly interesting and engaging. We do have a few questions. One of them is from the audience. Thank you so much for the talk, Professor Sirota. What do dimensionality reduction plots aim to do?
Great question. So we use dimensionality reduction to visualize data on kind of through a bird's eye view. So it's meant to take a highly dimensional data set that has maybe, I don't know, 20,000 genes or thousands of microbial species. Or we've used dimensionality reduction plots to visualize clinical data too. I didn't show that today. But each dot then is a sample or a patient, respectively. And the idea is to see, well, for molecular data, you can see batch effects. You can see if there's some correction that needs to be applied to the data prior to analysis. And you can see signal. Is there obvious separation between cases and controls or other features that you might not want there to be separation for, like batches, or let's say you can visualize demographics, age, sex when appropriate. So these are the types of things that we use dimensionality reduction for. Great question.
Thank you. Another question referring to the first part of your presentation. Did you encounter any drugs or supplements that may be contraindicated using these methods that you employed?
This is a great question. We could look at that positive score. So which drugs or compounds might be positively associated with the preterm birth signature. And we honestly, we have not looked at that. It's something to think about is in some cases, the effect that you see, the expression effect in a disease phenotype might be the body fighting that disease. So the positive relationships between the drug signatures and the disease signatures are kind of more of an unknown to us. So we haven't explored that systematically, but that is a great question, and there's an opportunity to explore that more.
Thank you. And Judith raised her hand. Let me see if you can unmute yourself. 1 second. Go ahead and you should be able to unmute yourself. Okay.
I see there's a few more questions.
Yes.
Oh,
Yes, I just saw that. Sorry about that. Yes, great presentation. I have background in data and analytics. How can someone with my background get into data science?
well, I have a feeling that data analytics is data science, so I think you're in it already. But I will say again that one example that I mentioned using AI tools for coding and developing models, I think can really bring people into the field of data science faster and enable that training. I mean, in my lab, we cross-train. So Brian, who I mentioned, led the drug repurposing work. He's a physicist by training. So he came to my lab to learn the biomedical side of things. He was very proficient in coding and math at the time, but he didn't know much about biomedicine. So we cross-train. Similarly, I've had physician scientists in the lab who want to learn and get into data science, and we cross-train in that direction also.
Awesome, thank you. And then do you think that there is a role for implementing OMAP to help with transporting models?
Absolutely. So for those who don't know, OMAP is a data modeling framework for clinical data, and all of our clinical data is OMAP. And the reason we've been able to port models from Stanford to UCSF and the other way around is because both clinical data sets are in OMAP. So yes, for sure.
Thank you. And one more question. How do you account for hallucinations from GPTs, AI in general?
Absolutely. Well, we look at many of them. So as you saw, we tested 8 different models, and you can really play them against each other. Well, there are several ways to do it. One way is to play them against each other and cross-check. Of course, regardless, in my lab, we do human code review. So that study, yes, so we tested 8 models. 4 of them were able to generate code that the students could run. But when we do discovery and students use these tools for code development, they still do human code review. And that's essential because not only GPTs hallucinate, we all make mistakes. So it's very important to do that. And also, all the code then is on GitHub afterwards. So the whole world has access to it. We're very open.
Very good. Thank you. And one last question, because we are right around 10 o'clock. What kind of governments or infrastructure choices at UCSF have proven most critical to making the data findable and accessible and still protecting patient privacy in cross-institutional studies?
So I can talk about the clinical data sharing a little bit. And basically, the data is de-identified. We cannot share the clinical data outside of UCSF or for the UC-wide, with the exception of the UC-wide kind of governance, where all the UCs are under a single IRB, which allows that cross-institutional sharing across the UCs. But we still can share models, though that now is being questioned with generative models or foundation models, which are very different than like a random forest model, a predictive model. So, for instance, for the collaborations that I've talked about with respect to Vanderbilt and Stanford, under an MTA, we can share the results back. We can receive a model, we can share the results on UCSF data. We cannot share the clinical data at UCSF with other institutions. So that's the balance that we have found. And then for all the researchers at UCSF, as I mentioned, everybody has access to the de-identified data. There's no IRB that's needed for the de-identified records, but everybody does training and attestation that, of course, nobody will try to find and re-identify the patients.
Very good. Thank you. Well, thank you very much. This brings us to right at 10 o'clock. And this was very engaging and very just fascinating. Thank you so much.
Thank you. It's my pleasure.
Thank you. And we have all three of our panelists here. Yes, we do. Wonderful. So let me rearrange my screen a little bit. Sorry, I have many screens open. Going back to, hopefully you can see my PowerPoint. Make sure. Sorry. All right. So it is, we are right on time at this point. We will begin our Ready, Fair, Go, making clinical and biomedical data findable, accessible, interoperable, and reusable panel discussion. We have three wonderful panelists today. I will introduce them one by one, and they will have some time, a few minutes to talk about their research. And then I will facilitate a panel discussion. We do, as I already mentioned, we do have some pre-prepared questions to get the conversation going, but feel free to drop your questions in the Q&A box, and remember to upvote questions as if you would like to get them answered faster. With that, I would like to introduce our very first panelist, Dr. Teresa Zayas-Caban. She is Assistant Director at the National Library of Medicine and Senior Advisor with the Lister Hill National Center for Biomedical Communications at NLM. Dr. Zayas-Caban was previously Chief Scientist at the Office of the National Coordinator for Health Information Technology, where she led ONC's scientific efforts and activities. In that role, she developed, established, or recommended scientific policy to the national coordinator, directed ONC's precision medicine initiative activities, and provided oversight of ONC's patient-centered outcomes research projects. Prior to her position at ONC, Dr. Zayas-Caban was the Chief of Health IT Research and Acting Director of the Division of Health IT at the Agency for Healthcare Research and Quality. While there, She set new directions for their funding opportunities and coordinated with federal partners such as the National Science Foundation. Before joining AHRQ, she served as a postdoctoral trainee in the computational and informatics in biology and medicine program at the University of Wisconsin-Madison. Dr. Zayas-Kaban obtained her doctorate in industrial and systems engineering at the University of Wisconsin-Madison, where she was a National Science Foundation Graduate Research Fellow in industrial engineering. And with that, please help me welcome Dr. Zayas-Kaban, and I will stop sharing so you can.
All right. Well, thank you very much. Thank you for the opportunity to be here. I will jump in quickly to my presentation. Here we go. All right. So I thought it would be helpful to provide a little bit of background in terms of how I got to my role at NLM at NIH presently. My background, as you just heard, is in industrial engineering, and I got into consumer health informatics as an area of application, specifically focusing on human factors issues, interested in finding a way to help individuals take care of themselves through the use of information technology. And I spent a lot of time in people's homes understanding how they manage health information and undertake health information management tasks to inform design of solutions. Through my role at the Agency for Healthcare Research and Quality, I expanded into clinical informatics. And once I shifted to the Office of the National Coordinator for Health Information Technology, really started to focus on putting health information technology to work for research and clinical research informatics, which brings me to NLM and NIH. I would say that overall, I've had an interest in building better systems, understanding how people perform health work and related tasks and use health information, and how that informs design and implementation of information systems in healthcare. And ensuring effective implementation and use, thinking about how we integrate systems into existing work, how we rethink work or support better ways of work using those systems, and of course, evaluation and continuous improvement, which is part and parcel for industrial engineering. And so the work that I've had the opportunity to lead and collaborate in really sits at the intersection of research and care delivery. At NIH and the National Library of Medicine specifically, our interests in data sharing and data science are at that intersection. Our goals really are to enable scientific discovery of diagnosis and treatments and new ways of delivering care, and to serve as a catalyst for innovation, both by making available data that can be presented in novel ways to researchers as well as providers, patients, and caregivers, and supporting novel delivery models. As the keynote speaker mentioned, the increased availability of electronic health data has certainly created new opportunities for health research, and it is important to determine how to best leverage the health IT infrastructure in support of the biomedical research enterprise. And so while I was at the Office of the National Coordinator for Health Information Technology, I was fortunate enough to lead a portfolio of projects that attempted to advance the use of health IT and electronic health data in health research. These projects span precision medicine, patient-centered outcomes research, artificial intelligence, and workflow automation. And I am going to focus on one, which was a policy and development agenda that we worked on. You see little numbers on the screen. I will share my slides, and at the end, there's sort of a list of resources for more information about these projects, should you want to delve into any one of these areas more deeply. And so one of the specific projects that I led resulted in this agenda meant to advance the use of health information technology and electronic health data and research. This consisted of a collaborative and multi-step effort that included a literature review, an environmental scan, workshop, multiple presentations with different communities, and which informed the agenda. As you can see, the vision that resulted from this effort is listed on the slide, really focusing on transforming the health IT infrastructure to support alignment between the clinical and research ecosystems to enable quicker and more efficient research. The agenda was organized into two overarching goals, one focused on electronic health data and the other on health IT infrastructure, with 9 associated priority areas really aimed at modernizing the nation's health IT infrastructure to enable research and discovery. These are areas that continue to be of great need, and I'm sure our speakers will comment on some of the things that they've been working on to make progress on some of these topics. More broadly, though, shifting to priorities at NIH and NLM, as you're well aware, scientific discovery is critically dependent on access to data. Biomedical research requires access to data types from diverse sources, including prior research. And the biomedical research community has really coalesced around the importance of making research data, findable, accessible, interoperable, and reusable or fair. And while advancing data access, sharing, use, and reuse are critically important to research, so is ensuring that the data are readily findable by those who need them. This is a priority both for the agency as well as NLM, as articulated in NIH's strategic plan for data science. The agency has prioritized data science to help store, manage, standardize, publish, and share the vast amounts of data produced by biomedical research. Additionally, in our strategic plan at the National Library of Medicine, we have not only prioritized access to the products and processes of scientific research, making them widely available, but also propose technical and scientific advances to ensure that research data are fair. And so NIH has prioritized providing access to findings from federally funded research. This requires a combination of NIH-wide policies, products, and resources or tools to do so. And efforts are advancing on 3 thrusts. The first one, ensuring that scientific data are generated from NIH-supported research are freely available as appropriate. Accelerating access to peer-reviewed scholarly publications resulting from that research, and identifying ways to support scientific and research integrity by enhancing collection dissemination of important metadata. And NIH has established different policies related to sharing of results from funded research, as you can see on the slide, and the focus on different areas. Data sharing more broadly, which was mentioned earlier in opening remarks, sharing of large-scale human and non-human genomic data, registration and submission of results for clinical trials, and providing public access to publications resulting from NIH funding. An additional way of ensuring results and data are accessible and findable is by making them available through different repositories and databases. As you may be aware, the National Library of Medicine manages biomedical information services, including such repositories. And some of these also support implementation of the policies that I just mentioned. So PubMed Central, for example, makes NIH-funded research publications publicly available. Clinicaltrials.gov makes information about clinical trials and related results publicly available. In addition, we make tools and resources available. So the dataset catalog makes it easy to search for datasets across a number of repositories. And the Center for Clinical Observational Investigations is sharing information on available clinical datasets to make them easier to use in research. And then Another pillar of this effort really is advancing the use of standards, data, metadata, and exchange standards to ensuring interoperability and reusability of data, allowing them to be more easily analyzed, shared, and combined with other data. This is another way to help us achieve our goals in data science at NIH through coordination and leadership by the National Library of Medicine and the Office of Data Science Strategy. We've been encouraging biomedical researchers to adopt and use some of the data and data exchange standards that are used in healthcare. And in addition to that, as you may be aware, NLM has a role historically in supporting standards by developing and maintaining taxonomies, funding some of these standards, doing testing and researching, maintaining relevant repositories, and supporting the use of standards in some of our information services. One point I really wanted to make sure I highlighted is the importance of collaboration in all of these efforts. Data science and informatics, for that matter, are interdisciplinary by nature, requiring collaboration amongst many different fields. My background's in engineering. Obviously, we collaborate with folks in medicine and nursing, library science, computer science, business, biology, math, and other disciplines. And similarly, it's important and very critical to engage relevant communities in all of these efforts to be able to effectively advance the use of biomedical and clinical data and research from patients to consumers, clinicians, standards development organizations, and others. Before I wrap up, I did want to mention that we have research training opportunities at NIH. There's summer internship program, applications close next week, and I have colleagues at NLM who are looking for both interns and postdoctoral fellows, so wanted to share those links. And so with that, I think I'll conclude and turn it back over to you.
Thank you. I think I am unmuted. Yes, thank you. Thank you, Teresa. This was very informative. And if you have questions, please put them in the Q&A box. We will answer them once the 3 presentations are done, and then the panel discussion starts. Quickly go back to sharing. All right. Okay. Hopefully you can see my screen. Yes, you can. Okay, so at this time, I would like to introduce our second panelist, Dr. Ashley Griffin. Dr. Griffin is an instructor at Stanford University Division of Computational Medicine and an investigator at VA Palo Alto Healthcare System Center for Innovation and Implementation. Her research focuses on developing and evaluating tools and methods to advance health data access and interoperability. Her work centers upon empowering patients to undergo and leverage their personal health data to improve their health and shared decision-making with care teams. She works with various data sources, including patient portals, EHRs, and patient-generated health data. Dr. Griffin received her PhD in health informatics from the University of North Carolina at Chapel Hill in 2021 and completed postdoc training at Stanford and VA Palo Alto Healthcare System Center for Innovation and Implementation in 2023. In 2025, she was awarded with the VA Health Systems Research Career Development Award to develop a novel patient-centered clinical decision support tool that collects, harmonizes, and visualizes patient-generated health data to facilitate shared decision-making and treatment opportunities for patients living with chronic pain. She also instructs Stanford's Modeling for Biomedical Systems course, which focuses on methods for creating computable, machine-actionable models of biomedical information so that knowledge can be shared and reused across different applications. So please help me welcome Dr. Ashley Griffin.
Great. Well, thank you so much. Let me go ahead and share my screen.
All right.
Right. Can you see this okay?
Not yet. We can see you.
Okay, not yet. Let's see.
It's happening, and yes, we can see it now.
All right, great. Well, thank you so much for the introduction and also for the opportunity to talk to you all today and tell you a little bit about my research and the work that I've been doing. So I'll be discussing about patient-generated health data, how we can make that fair through interoperable clinical decision support. So you've already heard a little bit about myself and my background, but my work really centers around shared decision-making, which incorporates clinical input and evidence both from care teams and clinicians, as well as from patients around their preferences, their values, and patient-generated health data. So I work in various domains here based on my background in informatics and consumer health informatics specifically. So I focus on data access and interoperability, being able to exchange data across different systems, especially between mobile apps and electronic health records. I develop and evaluate clinical decision support tools, both patient-facing tools that can be deployed through a portal or a mobile app, as well as clinician-facing tools that can be embedded within the point of care, within the electronic health record. Integration of multimodal data. This is especially important for patient-generated health data because it comes in a lot of different formats related to unstructured data like free text, reported symptoms, some structured data, raw accelerometer data, imaging data. So think about how we can integrate that together, especially how AI or generative AI can help us to do this. And then explainable predictive models that can be used at the point of care to facilitate the shared decision-making process. So that's a little bit more about my background. So patient-generated health data, if you're not familiar, is data that's created by patients or their caregivers outside of clinical settings to improve health or wellness. And this data comes in a lot of different formats and data types, including patient-reported outcome measures on symptoms, side effects, pain, functional ability, quality of life, and many more dimensions. Vital signs such as blood pressure, heart rate, weight from medical devices, as well as lifestyle and Fitbit trackers, for example, Apple Watch, and then other types of behaviors that could be related to physical activity, sleep, dietary information. And on the right here, these images capture different ways that patient-generated health data can be collected. And there's been growing evidence around the benefits of patient-generated health data, both for care teams and for patients. So these types of data can help improve our understanding of a patient's health in between their visits, which may be infrequent to the clinic. It can help empower patients to engage as an active partner in their care, identify meaningful health trends over time, such as being able to longitudinally track a person's weight or their blood pressure from home versus doing that in the clinic less often. It can help contribute to early identification of problems with the treatment plans or lack of treatment adherence or medication adherence and help to intervene earlier if we know what's going on. Supporting shared decision making. And this, of course, is very important for chronic conditions to help align the wide array of treatment opportunities with a person's unique experience and additional factors related to their lifestyle and behaviors and preferences. And then at a broad level, patient-generated health data can advance research and discovery by improving understanding of treatment benefits and effects. So just some early evidence that can suggest some of these benefits of patient-generated health data. But lots of challenges right now with PGHD. So these data are complicated and there's lots of variabilities at the individual level and the population level. So lack of standardized, interoperable data definitions and formats when thinking about how we can share data across systems. For example, there may be some devices or some blood pressure cuffs that track systolic and diastolic blood pressure within one variable and others that track it as two variables and many different ways that you could capture blood pressure, just as one example. And there's often proprietary hardware or software that don't interoperate. Many organizations actually lack the infrastructure to receive and to store this data and try to figure out some of the logistics of how the data gets stored, how it gets included within clinical workflows. And it's really important that we define meaningful distinctions for clinical measures and ensure consistency in how these data are recorded if they are to be used within clinical care. So thinking about distinctions like, is glucose fasting or is it non-fasting? Is the heart rate measured at rest or during exercise? Is physical activity self-reported or is it more objectively measured? And many more challenges here, just highlighting a few. So we can use the computable biomedical knowledge principles, or these CBK principles, to help us understand more about these challenges and possibly to overcome some of them. So these principles focus on a continuous learning cycle between data, knowledge, and a learning healthcare system community that leverages scalable, robust, and technical infrastructure. So in thinking about these CBK principles and applying them to patient-generated health data, in the first principle, CBK should be fair. So for findable patient-generated health data should have metadata labels. So thinking about who generated it, when it was generated, how, what device, and for what purpose. We also want it to be discoverable within portals or patient-facing tools and within the EHR. Accessible, so patients and care teams should be able to access, download, and share PGHD in an understandable, accessible format. We want PGHD to be interoperable, where we have standard formats and vocabularies. And then reusable in terms of provenance and being able to track what's happening over time, have appropriate context and quality indicators. So lots of considerations in terms of FAIR for PGHD. The second principle is around how consumers need to know how trustworthy the knowledge is. So where did the knowledge come from? Is it what I think it is? Is it being used in the right way? So for patients, we can think about how they need to know about the accuracy of the device or the app, the tool they're using, whether that data is clinically validated and what the data can and maybe can't be used for. And then for care teams looking at the data, they need to understand some aspects around data completeness, if the device has been validated, like from the FDA, and then how it might align with clinical standards and decision making. And then the last principle is CBK and its infrastructure should be open. So we want knowledge to be as open as possible to ensure equity of access to that knowledge. So we can think about having open standards like data formats, APIs, and terminologies. So with PGHD, for example, when we think about measurements like measuring steps or blood pressure, glucose, we could use LOINC codes. For symptoms and conditions, we could use SNOMED CT. And then FHIR, which was already mentioned, is an open standard for data exchange. And then also reference implementations and examples and documentation of how all these standards work together. There are a few implementation guides that exist out there for PGHD. So FHIR, if you're not familiar, is an open standard for exchanging health data so that different IT systems can talk to each other. It's characterized by a REST model for requesting and retrieving the data. It has resources for standardizing the data content. So we need to know what kinds of health data exist and how we can kind of group them together, like through patients, medications, lab results, symptoms, device readings, and have that data structured in the same way so that we could transfer it across different systems, health systems, or across different tools. And then OAuth for authorizing access to the data. You can think of this as like the sign in with Google. And so FHIR is really important for a lot of different reasons, but for this application of patient-generated health data, it's very useful for accessing the data through apps and devices and to be able to integrate the data within care settings. So SMART on FHIR it stands for Substitutable Medical Apps and Reusable Technologies, and it's built on top of the FHIR API. So it includes both authorization and authentication so that we know who is logged into the apps. And it's important for PGHD because it allows these third-party apps like mobile apps or a Fitbit to connect to an EHR system. So if we look at this visualization here, this example on the right here, let's say that we have a Smart on FHIR app that is within the EHR and it needs to access patient-generated health data. So it needs to get authorization from the patient in this case to some kind of API access to a FHIR server where this data holder is. And then it needs to be able to securely transmit that back to the app to be able to read from that data so that the clinicians can use this information at the point of care. And so the initial vision actually of Smart on Fire was developed based on this notion here on the left of how this traditional EHR is limited by the tools and the data that are developed as part of that EHR. Whereas on the right here with API-enabled EHRs, these enable third-party apps such as for remote monitoring or medication management, precision oncology, many different types of applications that can enhance clinical decision support and decision making and be reused across different healthcare systems here. So we can think about this as like creating an ecosystem of apps that can be reused across different electronic health records and different health systems. And so recently, in the last few years, there's been a new paradigm within clinical decision support of patient-centered CDS that incorporates patient-centered care that's personalized, proactive, and patient-driven, that's incorporating a family and cultural preferences, a person's values and goals of their care, collaborative care, as well as self-care, self-management, and skill building. And then linking that with CDS, or tools that inform patient care decisions of having the right people, the right format, the right channels, the right time, and then delivering the right information. So kind of linking these two domains together, we have patient-centered CDS, and these are both patient and clinician-facing tools that incorporate outcomes and measures that are meaningful to patients to inform care decisions. So this can include different types of patient data, including patient-generated health data, their preferences, goals, social determinants, and other types of data, evidence-based content, including evidence-based practices, alerts, and then visualizations like graphs, charts, and info buttons that can help us to understand and synthesize the information. And they can be deployed through different modalities for patients. It could be through patient portals, apps, wearables, or devices. And then with providers, it could be deployed through websites, but ideally within the electronic health record. So that brings me to Path for Pain Management, which is a CDS tool that I'm developing as part of my VA Career Development Award. And it stands for Patient Generated Assessment Technology for Improved Health. So the overall goal of this tool is to enhance pain management by building a shared decision-making tool that's collecting patient-generated health data and harmonizing it with EHR data to improve the use of non-pharmacologic pain treatments to reduce pain interference and opioid use. And so the focus here is on pain management initially, but really thinking about broader reuse and how this could be a framework for integrating patient-generated health data into the medical record for any type of chronic condition or for multi-morbidities. And it's actually a two-part tool. So there will be a clinician-facing tool and a patient-facing tool. I'm initially focused on building out this clinician-facing tool using that Smart on Fire framework. I'm doing this within the VA, and the VA is actually transitioning to another electronic health record, but will still continue to have their existing electronic health record probably for the next few years. They have a very long rollout plan. And so also have to think about developing this tool in an EHR agnostic environment so that it could be deployed through CPRS, their current EHR, and then Cerner, or I guess Oracle Health now. Also that this could be reused across any type of electronic health record, Epic or others that exist within other healthcare systems. So I won't talk too much about the details of this framework. I can go into more of this, but essentially linking different data sources together. The VA has a new patient-generated health data database that's stored as FHIR resources, and then different analytic environments. There's an operations environment and there's a research environment, and then kind of putting this together into a user interface, the Smart on Fire dashboard, harmonizing different data sources. So for example, the pain scores would be reported, medications would be in the medical record, non-medication treatments like cognitive behavioral therapy, that would be in the medical record, and then steps like from a Fitbit here, all anchored along the same timeline. These are the aims, very kind of traditional human-centered design aims. So just putting them out there in case anyone has any interest in these. So focused on gathering insights from veterans with chronic pain on their preferences for reporting their pain experience and goals and learning more about what's important for veterans. Using different methods here, some design methods as well as some NLP to extract preferences. Second aim is focused on clinicians and iteratively designing and developing the tool using that Smart on Fire framework. And then lastly, doing some usability testing and optimizing the acceptability and use with clinicians. So I think I might have gone a little bit over time, but yeah, I'd like to acknowledge a really big team here of collaborators, mentors, my project team. This is definitely a team sport. Yeah, lots of folks here from different backgrounds, also working with operational partners on this work. And then of course my CDA, which I'm about 1 year in. And yes, please do reach out if you have additional questions or would like to learn more. Thank you so much.
Thank you, Ashley. That was very interesting as a patient that we all are at one point or another, and then your work at the Veterans Administration is just so very important. So thank you. I will, at this point, return back to my PowerPoint and I would like to introduce our third panelist, Dr. Daniela Meeker. Dr. Meeker is an associate professor of biomedical informatics and data science at the Yale School of Medicine and the chief research information officer for the health system. Her work includes high-impact field experiments applying informatics and behavioral economics, engineering systems for data integration and analysis, and contributions to data policy and standards development. Please help me welcome Dr. Daniela Meeker.
Thank you. It's so nice to be here. Thank you for inviting me to be on this amazing panel, which has Teresa, who's a rock star in our field, and Ashley, who's clearly a rising star. I feel really excited to be able to present with them. Going to try to share my slides. Here we go. Bear with me. Okay. Put this in presenter mode. Does this look the way that it's supposed to look? Great.
Yes, we can see the presentation.
Okay, yeah. So thank you guys again for inviting me. I put together a few slides, mostly oriented around trying to provoke some discussion for our panel around some of the tensions between efficient operations and the process of promoting interoperability and standardization and the things that are needed for effective data sharing. And that's very much in light of the fact that I wear a couple hats, as you heard in the introduction. So I have an operational hat where I am responsible for managing the overall research informatics program for our health system and our school. And I also do some of my own research. I eat my own dog food when I'm trying to do things that are in the realm of pragmatic trials and implementation science. And I just wanted to highlight some things that all of the earlier talks have sort of touched on, so I won't have to go into agonizing detail about them, but really shining a light on the difference in priorities between the healthcare operations and the research aspects of those operations, and what we're doing in research networks and individual research projects where there's maybe more of a foregrounding of this kind of data sharing activities. So really, when we're trying to implement these kinds of research operations and get them to be embedded in health systems, the focus is on trying to coordinate with some existing systems that are in place. Very rarely do we get to be involved from the ground up. And many of the incentives for standardization are not fully recognized. Even things that going back to things like meaningful use and different kinds of pay for performance programs, in practice, they often end up being not fully realized the way that ONC envisioned them when things trickle down to the front lines of a healthcare organization. And so a lot of the things that we can do as researchers is try to build the bridge between some of those policy visions and what's actually happening on the front lines, but rarely are we available or involved in the kinds of situations where someone is making a big procurement decision, for example. There's also a fair amount of agility, at least in the healthcare systems that I've worked with. I've worked with public systems, private systems, academic systems, and there's a lot of competing priorities that have to be managed if you're embedding research into the health system. And much of the interoperability is somewhat the minimum necessary. And so all of that impacts the quality of the data that we have available to apply in, say, a clinical decision support application through to secondary analysis of the data and AI. So I think when we're working, especially in multi-site studies and research networks, there's more of a foreground on the interoperability. The collaboration is built in from the ground up, and having these kinds of standards are essential. I really liked Marina's slide that sort of showed the different UC systems and how you can go really deep with one system's information, but when you're trying to share across multiple organizations, I believe they're still using OMOP for that project. They have a little bit less of a deep and more of a broad perspective on what's happening, and that's sort of part of the negotiation that's happening in these kinds of standardization activities. So that's sort of the sort of tension that I'm trying to navigate in my day-to-day work, and also when trying to sort out ways to work together across multiple organizations and promote activities that will advance fairness in the learning health systems. And this is a diagram that I stole from one of Chuck Friedman's websites, the Learning Health Project. And I love it because it spells out all of the different components and how the work we do really fits into this. So I think, with this first sort of portion of the wedge, trying to drive interoperability between research information technology and health information technology, fairness is very central to that, making sure that we have a really well-informed way of adopting and developing standards. And then as you go into having more and more information that could be applied, you build into this second wedge here, the knowledge to practice flow and how knowledge is represented. So some of the things that NLM has funded are related to knowledge management and how these knowledge graphs can actually be applied, as you saw in Marina's talk, applying these knowledge graphs where you have information about the pathways to advance in discovery throughout the research life cycle. Those are applications of things that have required interoperable knowledge management. And this is sometimes overlooked, but those knowledge graphs, especially when we're talking about the NLM assets, are incredibly powerful and in some cases, underutilized when we're talking about learning health system, decision support types of activities. There's a lot of opportunities there to take advantage of that space so that the knowledge actually is continuously maintained. And one of the things that we've been working on in our system is figuring out, how do we do that stewardship of knowledge so that the latest information, say on pharmacogenomics or the contraindications, are really kept up to date and integrated into the day-to-day practice of our clinicians. Also looking at standards-based consent and data capture. So we've talked a little bit already today about patient privacy and how we can capture preferences for data sharing at the patient level. There's been work with the FHIR standards on very granular consent information, not only specifying what attributes can be shared, but even with whom they should be shared. I think that will continue to be an emerging important space as we get more and more availability of third-party applications for decision support and precision medicine. So I think it's often overlooked, but it's quite important when we're talking about trying to bring these applications forward and make collaborations across different entities. So the research that I do is on this bottom part of the wedge, so the practice to data flow. So we can look at inducing variation in practice with pragmatic trials. So that's when you're doing some kind of a nudge to change the adoption of a particular treatment or a particular tool and do the downstream evaluation of the impact of that. So that's when we have sort of a proactive opportunity to intervene. However, sometimes there's already practice variation in adoption of these types of things. That's the result of a natural experiment. One health system implemented something one way, one doctor has adopted this new treatment while his colleagues have not. Those types of natural experiments and monitoring for unintended consequences of policies or apps, they can also contribute to this knowledge base. And so all of these things work together ideally in a virtuous cycle that are integrated with different types of technology platforms. And that kind of big picture does a lot of glancing over the trade-offs that we have with standardization. So I love this quote by Bette Midler about how we should really be focusing on what are our minimum standards, because there's always going to be some kind of investments at the time of implementation and data collection, unless we want to go forward with post hoc standardization. So you can invest up front, you can invest at the end. At some point in the process, you're going to incur those costs if you want to be able to combine data and assets across multiple sources. And that often requires subject matter expertise, maintenance, but it does then have the benefit of generating these reusable resources that can readily be interoperated, not only across different data sets, but also with these different knowledge assets that you can hit against the data to make different types of inference. And finally, what types of things require relocalization when you're translating some component of knowledge, say the information for a decision support rule, to translate that into another site from the original place it was developed, you may have to relocalize it. Again, like this perfect example of the difference between the two UC systems, where they just captured completely different information. So in order to make their decision tool interoperable and functional, there had to be a re-estimation of the model based on what the codes were that were available at the different sites. So that's always something that we have to keep in mind when we're thinking about what the minimum standards are. So the next couple of slides are sort of what I think might be opportunities for us to discuss. I think this community is such a great consortium for bringing up different types of things that are available for us to collaborate on and share ideas on. The different kinds of standards that have been widely adopted in the research community. OMOP has been a pretty broadly adopted standard, in part because of investments that the federal government has made in initiatives that have adopted this. I actually haven't listed all of them, but programs like All of Us, NIA-funded networks, the Odyssey Consortium is a fantastic collaboration that has both federally funded and privately funded projects that have been dependent on adopting this standard, which is very much based on the HL7 terminology resources. More recently, there's one EHR vendor, and I don't want to shill for them, but they've created a community standard for one of their data models, and it's enabled a fair number of projects across their different clients, especially academic medical centers, including a very large foundation model. And thinking about how do we go back and forth between different standards or community-based standards to achieve the same kind of portability of knowledge and analysis is part of what I think we can work on together. I brought up imaging. We didn't talk too much about imaging today, but one of the areas that has been a real barrier, going back to this patient privacy issues, is how we can share images. And if we adopt some of the basic standards, which DICOM is very widely adopted, that has enabled a lot of AI to be developed, as well as things like coordinated de-identification. And there's a lot of caveats with that, but that's kind of an area that I see interoperability as being fairly successful actually in the research and AI space. And then I'm just highlighting some work that's been done at both Yale and at USC in automation. And given that this is may have some library sciences audience, automatically doing indexing and metadata extraction from poorly curated data sets has been explored with a couple of different tools. And I'm just going to shout out my colleague Hua Xu, who has built a tool that applies large language models to automatically pull the data together and harmonize it, which may lower the barrier to standardization across these different projects. And finally, I just have a couple sort of proposals for us to discuss. So there's been 3 or 4 very large foundation models based on electronic medical records. And what I mean by that is we have now seen several different organizations that curate sequences of medical records, translate the same methods that are used in language models. So instead of next word prediction, it's the next code prediction. What is the next ICD-9 code? What is the next treatment? And those projects, unfortunately, haven't been based on any standard approach to tokenization or embedding or even the source data. So of the models, one is based on, of the large models, one is based on UK Biobank, another one is based on the MIMIC data set, the other one is based on Epic's proprietary model. And I think there's a real opportunity to be able to apply these models and explore what are the methodological limitations compared to the data limitations. Right now, because they're not interoperable in any way, we don't know whether it's a shortcoming of the way the data was embedded, the way the source data varied, or if there was some special sauce in the methodology. And that even goes all the way to evaluation. We can't actually test the same validation question on 2 of these different models. And some of your colleagues at Stanford have pointed these kinds of things out. Nigam Shah has written a few papers about some of these issues. And I think that there's a lot of important points that have been made that should drive us towards some more interoperable approaches. And I put a star on this last point because some of the standards that have been developed, long-standing standards that have been developed for electronic clinical quality measures, actually rear their heads again when we're talking about using these large data models or sequence medical event models, because just like a clinical quality measure or a clinical decision support rule, you're prompting these models with a sequence of events that happen over the record in somebody's chart. And that's exactly what electronic clinical quality measures and other CQL-based artifacts are doing. And I think that will probably come up again in a way that might have been unexpected with these AI tools. Other areas where there's not a lot of standards, but a lot of opportunity for collaboration. The audit and access log data in our electronic medical records. There's a lot of interesting metadata in there that can pertain to user experience and the patient experience that we haven't really done as much analysis on as we get into these kinds of adoption and evaluation of decision support tools, like what Ashley talked about, understanding what the uptake and utilization is requires using this data. And we don't have a standard way of evaluating that. And I think I'll just point out that there's already been several collaborations across the UC system and Stanford looking at ambient listening, but we have not come up with some type of standard for how to manage the information that's based on recordings of encounters between patients and their doctors, ranging from the acoustic data to the lexical analysis to the ways of representing the different content in notes, and how we might do dimensionality reduction on those topics. The last thing I think that we might consider is how we can lower the barriers to achieving interoperability across our different systems. So the traditional way of doing this is by hand tooling the mappings and hand creating the business rules. But there's been an increasing number of AI assisted methods for doing this, and one type is just side by side with your AI tool. Does this term map to that term, and how do I validate that with a subject matter expert, while still like mostly manually developing these kinds of logical and mechanical transformations that have to get done. But the way of the future might involve even less of a human in the loop and more of a supervised learning with a fine-tuned model that is doing things like evaluating based on things like conformance checks. And so if we've brought up OMOP a few times, there's a library of tens of thousands of conformance queries that model has, and you could easily imagine training a model to do the standardization from the VA's electronic medical record to OMOP, and as they have done in Vinci, and try to compare a hand-tooled version of that to one that's been developed with some of these AI tools. And I think that may help us get closer to interoperability and lower some of the barriers. So I'm looking forward to our panel discussion. Thank you guys for inviting me today.
Thank you so much, Daniela. That was very interesting and brings up lots of questions. Some of them we are probably going to cover in the panel discussion. Please remember to put your questions to the speakers in the Q&A box. You can upvote them to get them answered faster. And I'll start with a question that's in here. It came in during Ashley's talk, but I think all of you can touch upon it. The question is, thanks so much for these great talks. When a predictive model identifies a high probability of a poor outcome based on a user's behavior, should the decision support tool be designed to prioritize guiding the user toward the statistically safest path, or presenting options neutrally even when the data suggests the user is likely to make a suboptimal choice? And anybody, feel free to unmute yourself and elaborate.
Yeah, I can kick us off there. I think it's really aligned with one of Daniela's slides towards the end about having a human in the loop, which I am a really big advocate for. And I think when any kind of decisions about outcomes are to be made within clinical care, that it is important that the model is tested and validated. And I think in this case, when it's kind of based on a user's behavior, that where there's different options about, if the user's likely to make a suboptimal choice, I think that it's really important to have a clinician, an expert there who's able to review those choices. So I kind of see it in different parts of like being able to ensure that the model is robust, that it's tested and validated, especially on different kinds of populations and the population of interest here, but also that a clinician is able to review it and then discuss those options with the patient.
Thank you, Ashley. Excuse me. Teresa or Daniela?
I think I might add briefly that an important avenue research is how to convey probabilities to users and do so clearly. So if that kind of information is going to be included with decision support or other kinds of predictive tools where, either to be used clinicians on their own or with patients or patients by themselves, we really need to think about the user base and how we convey this information clearly and ensure understanding.
Yeah, I agree with that. I think that even clinicians are not so great at interpreting uncertainty and probabilities. So I think that communication is definitely a challenge. I think in addition, the obtaining preferences and translating those preferences into a way that informs the predictions is a sort of a newer component of what a number of people have been working on in order to help convey sort of how whatever decisions are aligned with the preferred outcomes, and even understanding how those preferred outcomes map to those things is itself an area for research. So I think it's definitely an interesting question and brings up itself like several avenues that could be explored further.
Thank you. The next question is also from the audience. And I forgot to mention this. I will copy and paste the questions into the chat for the panelists to see, because sometimes they are a little bit longer. Is there any recommendation you may have for business concerns trying to validate with subject matter experts that have university constraints or communication. Sorry. I will drop this in the chat.
Let's see. So I think I'll try to take a stab at this. I welcome other panelists to talk about it. And I make sure I understand. I'm interpreting this correctly. So is the question about conflict of interest, and what can be shared publicly? Is that the gist of this? I definitely feel as though like having, navigating things like non-disclosure agreements as well as, conflicted interest between people that have their academic hat on versus, their business hat on has been an ongoing theme in the projects that we've been working on. And even, at what point do you consider the development of one of these tools a partnership with a business versus something where you're a client, and sometimes even those lines can be fairly blurred, and that's why most academic centers have a translational office of some kind. However, most of their work is oriented more around drugs than these kinds of decision support tools. So I definitely think that those navigating those types of issues continue to be a concern. But I hope I've answered the right question, because I can interpret it a few different ways.
And yeah, I guess for me, I can weigh in because I work both at a university and a healthcare system. And I mean, the only thing that I'll add is that working at the healthcare system, it's really important to get buy-in and input iteratively and over time on the model or the decision tool, because we want it to be valuable for all kinds of end users. And things can definitely change over time. I mean, now we're kind of in this new era of generative AI, and so people are thinking more about that. But I think it's really just, important to get buy-in from the end users when kind of building these tools. I'm not sure, also if that's answering the question.
I'm not sure if this is a follow-up to the question, but in the Q&A it says outreach from external organizations. I don't know if that clarified it.
I would definitely say that we probably have 4 or 5 startup type organizations reaching out to our faculty and clinicians and senior leadership every week. Like it's a constant flow of people. Some of them are internal and some of them are external. And I think each of those organizations wants us to do a pilot of some kind. And we have sort of like thousands of these different proposals at different phases of acceptance, and it can be overwhelming, especially when you have to also take into account that some of these are initiated not exclusively by the company, but by also people that are, have faculty positions or clinical positions in the organization. The way that we have tried to solve this, I'll give a call out to Lee Schwam, who has put up a lot of governance around these types of things at our organization. I think many schools and academic medical centers have set up some level of governance around it, but they've sort of put some pretty clear lines on having some type of executive sponsor that is at the senior leadership level to filter what's coming through, so that there's just not an overwhelming amount of pilot work that's going on, because the implementations can be quite expensive, especially for new organizations. So how to foster that innovation while while making sure we don't get distracted from the high priority projects.
And speaking of innovation, the next question is, drop it in there. Looking ahead about 5 years or so, what emerging trends do you think will most transform how FAIR principles are applied in clinical and biomedical research, and why?
I feel bad that I'm like being the first one to answer these questions, but
Please do.
I'll jump in. I actually think that each one of these will have its own kind of impact on sort of the direction things are going. The federated learning has really exploded since the advent of more of these AI methods and parallel architectures. So a lot of the original algorithms for federated learning, they go back to like the early 90s when they had to parallelize things to implement neural networks. But there's been a huge amount of innovation since we have transformer-based approaches that I think is, it's in order for like truly federated learning to work, you do have to have an interoperable data set that is as as standardized as something you would put into a data commons. So in some ways, it has to be even more rigorous because you have to deal with these kinds of interoperability issues that came up of like, oh, is this data element even available across all sites, for example, or has it been translated in the same way? So that's definitely an important thing to keep in mind. I'm less enthusiastic about synthetic data in some ways. However, like these new generative models that are based on the foundation models for electronic medical records, they will create a huge amount of synthetic data if necessary. Although there's some privacy issues that haven't been fully addressed there. So I think that there's a lot of privacy and security research on what gets revealed when you have these kinds of generative models that have been trained with real patient data, for example. And I think that because we don't know what the privacy implications are for these types of models, I'm a little bit less gung-ho until we resolve those issues about synthetic data, because it has that sort of problem that people are balancing. And yeah, I think that there's been surprisingly good success with these centralized data sets that have been based on consent. So all of us, UK Biobank, like these have been really successful projects. So they have their own limitations, of course, but they definitely have been high impact, high reward programs. I feel like Teresa probably has like another perspective on this too, coming from her perspective.
Yeah, I mean, I'd say not necessarily how the principles are applied, but more advanced. I'm excited to see innovation in automation of data curation. Some of what Danilo is mentioning toward the end of her presentation in terms of um, ways to more easily and quickly either derive meaning or annotate the data to facilitate quicker access and use and overcome some of these issues around interoperability and standardization. I think those kinds of advances will merely make a significant difference in making data fair.
Yeah, I think those were great responses. The only other thing I'll add is that LLMs are fundamentally changing how we do science and how we work and how we live and interact with health information. For example, now with I guess there's ChatGPT launched a health GPT where you can upload your entire medical record now to ChatGPT. So yeah, lots of interesting things happening within industry and things that are coming down the pipeline. Yeah, so I think it would be really interesting, and I'm excited for the future of applying these FAIR principles to LLMs, with ambient listening and how we can have standards for capturing that data, making data that's AI ready, that can be used by these generative models, and then having frameworks like with human in the loop to make sure that the data are fair and that we're applying them in the way that we had planned to.
Thank you. And kind of along those lines, and we are running a few minutes late, but I would like a few more questions if we can answer them. And going along with what you just said, Ashley, FAIR emphasizes reusability, but secondary use of clinical data can easily reinforce existing biases. How do you assess and mitigate these biases when preparing data sets for downstream machine learning or AI applications?
Yeah, I guess I can kick us off there. I think that there's a lot to unpack in this question and really important question there that's just kind of fundamental in any kind of machine learning and AI application is to make sure that you understand the biases and then opportunities to mitigate the biases. There are some frameworks out there that can help us to kind of guide it and to make sure that these models are interpretable. And that we can kind of reduce the downstream and perpetuating of these biases. So yeah, I would say that a point is really something that's central to everything that we think about. I mean, I'd be curious if others have kind of specific areas of how we can mitigate that.
And I definitely feel as if something that's very interesting about these like newer large language models is being able to query them for representativeness of your particular question in the training set. So that requires, a fair amount of documentation and even itself brings up some privacy issues. But I think that being able to navigate the same kind of uncertainty that came up earlier in the conversation is if you are not represented well in the training set, this model might not be one that is useful for the particular question that you're asking, and being able to advance more transparent information and metadata about the training set is probably a way to help us, at an individual personalized level, understand what kind of biases might be introduced there. And that those error bars around the response are sort of not one of the great features of our latest like chat-based models, but they are things that can be generated as we're thinking more about precision medicine.
All right. And last question, because we are running late-ish. For attendees who are inspired today and want to take one actionable step next week towards more fair clinical and biomedical data in their own work, what is the single most impactful thing you would recommend for them they do?
That's also, another great question. I think I would say something around documentation and metadata and making sure that everything you do is written down and properly documented in a way that you can easily find it, in a way that it can be reproduced. I mean, I think in science where people are often very critical of the work that you do. And so I think making sure that we align with FAIR principles and having the documentation, I think that makes things a lot more open and up to less criticism.
Yeah, I have to agree with Ashley to bake it in from the beginning and think about reusability and availability as you're planning out your work and not try to retrofit it or add it at the end.
Yeah, I'll throw into the crowd where I think, if you're not sure about how to do that, consult with your medical librarian, and they will definitely be able to help you develop a strategy for your project that meets those needs. So earlier consultation, better.
All right. So we have more questions in the Q&A, and we are running 7 minutes late at the moment. There's one for Dr. Meeker. Dear Dr. Meeker, I am a data scientist currently working in disease gene prediction tasks in collaboration with a genetic counselor. You spoke about AI algo versioning. How do you suggest versioning graph-based ML models when they continuously learn from updated biomedical knowledge graphs?
A great question. I mean, I think that this also goes back to the transparency and the representativeness of the data. So being able to maintain a continuous record of what the training information is, and some summary, at the minimum, some summary information about what's happening with each of the reweighting iterations are going through. It's almost like a parallel data set that you might have to maintain throughout that. It's a great question and a general problem, especially when you have very small tweaks over time that are happening. So I think if anybody here has found a good solution to this that doesn't basically require maintaining that sort of catalog of training data on a regular basis, I'd love to hear suggestions. And that is something that I think we're always struggling with, especially when, there's almost an infinite number of models that are being developed as people are fine tuning things when they go beyond like zero shot learning types of projects.
Thank you. All right, so I would like to thank all of our panelists for graciously sharing their time and expertise with us today. This was a really, really fascinating and inspiring session, and I wish we had another hour to continue the conversation and answer the questions. However, we do need to transition over to the next portion. So thank you so much for being here. I can't thank you enough. Thank you. And at this point, I had to change my slide while I was talking from a 5-minute break to a 3-minute break. We are going to take a very quick 3-minute break so everybody can stretch and get some coffee or tea or some refreshments. We will be back at 11:18. And at that point, I will introduce our final speaker, our closing keynote, Dr. Erica Johnson. So 3 minutes. All right, 3 minutes goes by very fast. I hope everybody got a chance to stretch and get some refreshments. At this point, we're coming back from break and we are here to listen to our closing keynote. It is my honor to introduce Dr. Erica Johnson. Oops, change the slide, there you go. Dr. Johnson is the professor of gender and society at Linköping University in Sweden. He is the director of the Swedish National Graduate School for the Wallenberg AI, Autonomous Systems and Software Program, Humanity and Society. Her research focuses on how data represents the world and objects they claim to describe, with a special interest in medical data. She has an interdisciplinary background in sociology, gender studies, and science and technology studies. Dr. Johnson is the author of several monographs and anthologies, including a cultural biography of the prostate, which was published by the MIT Press in 2021. She was an editor of She was an editor of How That Robot Made Me Feel, also by MIT Press in 2025. And she co-authored the recent Big Data and Society article, The Ontological Politics of Synthetic Data, in which she details intersectional hallucinations. She is a member of the Royal Swedish Academy of Sciences. Please help me welcome Dr. Erica Johnson. And I will stop sharing so you can share.
Thank you. Is my slide there now? I think
Yes, your slide is there and we can hear you.
Okay, great. Thank you so much for having me here today. It's I've been really enjoying the talk so far, and I hope that this keynote will trigger a few questions and maybe some confusion, because that's kind of what I hope to do. The official title of the talk is Synthetic Data and the FAIR Principles, and I am going to talk about that. But an alternative title to this talk could be, So You Want to Share Your Synthetic Data, Let's Talk. And the reason I call it that is because towards the end of this presentation, I'm going to detail the work that I've been doing with the Swedish National Data Service and the UK National Data Service around how data repositories, especially working within an open science paradigm, could possibly be thinking about principles like the FAIR principles and synthetic data. But before I get to that, it probably is wise to start defining what we mean by synthetic data. So I'm talking primarily today about AI-generated synthetic data. And I say that because you probably are quite aware that synthetic data, because you are data scientists, synthetic data has existed for a long time. We have had synthetic data as a standard practice in a lot of the census data that we encounter for many decades. And we have lots of domains of science, like, for example, environmental sciences, that use a lot of model-generated data, and that is also at some level synthetic data. But AI-generated synthetic data is slightly different, and I think it is doing new things to our needs for trying to create transparent and replicable data within any paradigm, but especially within the FAIR paradigm. So what kind of synthetic data are there? And what do we do with, how can we recognize them? Well, I think we are all familiar with the types of synthetic data that is image data, which is probably flooding your social media feed as much as it's flooding mine, and it's definitely flooding our news feeds on every media, and has been one of the more successful types of synthetic data, at least initially, because it was so important for the areas of AI development around autonomous systems, the self-driving cars, which needed to be able to train on a lot of images in order to train their models, and started to create synthetic images in order to do that. So this is one type of synthetic data. There's also all of the language that you get out, I say language in quotation marks, but all of the tokens that we read as language and interpret as some sort of information that any sort of large language model chat, GPT or something, will give us. It's also synthetic data. We interpret it sometimes as knowledge, sometimes as wisdom, sometimes as compliments, but it's just data, synthetic data. And then there's the type of data that I'm going to focus on today for this talk, which is tabular data. And tabular data is definitely not as sexy as like Trump as Pope or whatever ChatGPT is telling you about how smart you are, but it's definitely synthetic data, and there's more and more and more of it being encountered. And the panel brought this up a little bit today as well. So I think synthetic tabular data is starting to be something we encounter. We don't necessarily know that we're encountering it as easily as when we see some of the images that are also synthetic data, but it's there. So why would you want to make or use synthetic tabular data? And there are some pretty fundamental reasons for doing this that are problems in quotation marks or hiccups for many of us working with data that a lot of people try to get around using synthetic data. One of them is that synthetic data, at least so far, is usually pretty portable because it's not really governed by many of the types of data regulations or ethics approval regulations that we have in place for a lot of our other data. Maybe it should be, maybe it will be, but right now it's not. So it's definitely portable. And portable data, like we've been hearing all morning for you, is something that we want. Also, and here is a big maybe, and this was a maybe that was also brought up in the panel, which I was happy to hear, but maybe synthetic data can partially help us provide privacy for our data. The academics I speak with, I'm an academic, we're kind of not really sure that that's actually going to happen, but a lot of people will try to tell you that synthetic data can assure privacy for your data. It's a big conversation. One of the more prominent reasons for using synthetic tabular data is that even though somebody may have a lot of data and want to use it to train their AI models on, they probably don't have enough data to do that because they just need so much data for that. So synthetic data can allegedly be used very successfully to augment or amplify your real data set so that you have a large enough data set in order to train an AI model on. And then also, which was mentioned before, was that maybe, and there's this idea that maybe we could use synthetic data to balance out or address known biases in our existing data. Maybe it can be. So this would be another reason to use synthetic data. Now, I have to say, for all of those of you who are in the audience who are as old as I am, when I hear these reasons for using synthetic data, and arguably, I'm not a data scientist. I'm a computer scientist, or not a computer scientist. I'm a social scientist who has worked for a long time at the intersection of computer science and social society. But when I hear these reasons for using synthetic data, I am reminded of that 1990s film, Austin Powers. And I start to think, what are we really trying to do with our synthetic data? But I will leave that there, and I will go forward and talk a little bit about the synthetic data and what could possibly go wrong, given that there are so many reasons for wanting to use synthetic data. And I embed this in a bit of background. So I've been working with computer science and society, a lot of it around medical data and medical technology, science and technology studies. And as I've been doing that, I've been working a lot on how the body is represented in medical data, or how the identity or we as people are represented in medical data. And I'm a gender studies professor. So, I'm kind of interested in like minority groups or minority groups in the data, which is oftentimes women, even though we're not a minority group as such. So a few years ago, I was hearing within my medical humanities bubble, a lot of people in the medical domain saying, yeah, synthetic data is probably going to solve a lot of our problems. But I'm also in the AI domain, that bubble. And in that bubble, I was hearing people say, well, yeah, I guess maybe, but synthetic data does things. So for example, when you create synthetic tabular data with the tools we have now, you have a pretty strong chance that your synthetic data set is going to amplify the majority elements in your data set and minimize the minority elements in your data set. So basically what that means is if you think of your data as a bell curve, you're going to get a synthetic data set that has a lot of the stuff in the middle and probably not much on the ends. this is something that could happen. So 2 years ago, I managed to get a grant to work with a computer scientist and start testing some ways of generating synthetic data. And all of the examples that I'm going to show you now are results of those tests using population data. And we chose to use population data because it's easy to understand innately as a human what's happening in that data at the human level. But the things that I'm talking about will happen with any tabular data set. So just keep that in mind. It's just easier to understand with population data than, say, with deep sea currents and temperature and salination. So I'm going to give you some examples. So what could go wrong? Well, what we saw was that when we started generating our synthetic data sets, the edge cases went missing. And this is what leads to mode collapse with your AI models later on. So for example, in the 1990 US Census data that we used, because it's an open access data set and lots of people are familiar with it, we noted that in the original data set, there were 41 countries of origin. So the adults came from 41 different countries. Now, most of the people in that data set come from the USA. They're born in the USA. But there are people who have immigrated to the US, and those in the original data set had immigrated from 41 different countries that we were using. But in our synthetic data set, we only had 32 countries of origin. So 9 of the countries had gone missing. And these were the countries where there were very few examples of people who had emigrated to the US at the time in 1990, Hong Kong, Hungary, Ireland, Laos. Those were simply gone. And more to the point, we also noted that the relations between the different attributes, the different columns, the different things describing the people in the data set also changed. This was and is a result of the fact that the columns in a data set are actually saying something between them, but the models we use to reproduce and synthesize data sets are working primarily within the single attribute. They're working intra-attribute, but we're interested in the inter-attribute relations of the data set. This is very complicated. I'm going to give you some examples, but what I'm going to show is don't get panicked about the slide. I'll talk you through it. I'm going to show is that when we start thinking about data sets as more than just statistical representations, but rather representations of ways of categorizing the world, which is, thinking about these as like ontologies of the world for us, full of politics and power, then we start to see that the AI models that we're using right now are not necessarily going to be helping us all the time in the synthetic data set. So here is a SPLOM, a visualization of our original data set, 3-point intersections of it, and the synthetic data set, 3 points intersections. And then the diagonal along the middle is a single attribute comparison of the 2 data sets. So here, for example, we can see how age in the real data set and the synthetic data set compared. And we can see that the synthetic data set was pretty much had the same statistical distribution of age as the real data set. It wasn't identical, and that's good, because if it was identical, it would just be a mirror of our data set, and that's not what you want with synthetic data, but it was pretty close. And if you look at every single one of these squares in the diagonal, you'll see that we basically had a pretty good statistical representation of the real data in the synthetic data set. And that it was not identical, which is key, but that it was pretty close. And the reason it's key that it's not identical is because if you have a synthetic data set that is just a mirror of your data set, it will not be able to avoid data regulations or assure privacy, even if we think that it can do that. And it will definitely not be adding new information to your augmented or amplified data set in a way that could give you more things for your model to learn during the training process of your AI development. So you must have a synthetic data set that is different. So you want it to be different, but you want it to be pretty much the same. Okay. Fine. So at the single attribute level with this particular data set, we were able to get it to be pretty similar at the single attribute level. But then, okay, I'm a gender studies professor. I was like, talking to my colleague, Dr. Sagi Haji Sharif, I'm like, okay, but let's think about this. We're using the concept of intersectionality because we know that we are all much more than our age, right? We are also things like our gender and our occupation and our educational level. These all impact us as people, and this data set is actually a representation of people. So let's see what we can do when we compare them at sort of an intersectional level. So we decided to compare here, looking the age and income and gender of the data set, synthetic and real. Real being on the left, synthetic on the right, the slightly grayer one. And here's too, we can see that, okay, gender was the pink blue. This is 1990, so it was definitely binary. Income on the one axis, and then it was oh, goodness me. Gender, income, and age on the other. And we can see that these two data sets are pretty similar at this particular intersection, but again, they're not the same. They're not identical. So I don't know, maybe this is like a pretty good representation because you don't want it to be the same. You want it to be different, but you kind of want it to be the same. But then we look down here where we were looking at occupation, marital status, and gender. And here we saw that there was an awful lot more pink in the one in the synthetic data set. So we went back in and started to check out what was going on in the data. And we realized that in the original data set, we had one female husband. But in the synthetic data set, we had 259 female husbands, which today and in some countries, like in Sweden, totally fine. But if we're trying to synthesize the imaginary of the world that the data was representing in 1990, this was probably not a pretty good representation of it. So, and we also had, I should note, 333 points where we had like a person or a data point that was both either a husband or wife and also single. So this was definitely what you could call in machine learning language an implausible variation. I'm the social scientist in this team. So I was like, Soggy, this is amazing. We have to think of like words, terms, like theoretical terms that we could use to talk about this. Let's call it intersectional fidelity where it's good and intersectional hallucinations where it's bad. And so then I sat down, I started writing this paper about it, the one that was mentioned earlier on, and then we theorized a whole bunch about it. But then Soggy, who's also an engineer, she was like, okay, we were using GANs. Why don't we try to use a diffusion model, a different type of AI to do this work? And she did. And what she ended up doing was creating a more accurate synthetic data set where we had 68 points that were husband and wife and single instead of 333, right? And we had fewer hallucinations at the intersections that we were looking at, which is, great, I guess. But it also begs the question of how close do you want your synthetic data set to be to your original data set? Because again, if it's too close, you're not going to avoid data regulations. You will definitely not be able to assure privacy. And you're probably not adding that much more to your information to your data set if you're going to amplify it or augment it with this synthetic data set. So where on some sort of sweet spot balancing board of your surfboard with data is it that you want to have to balance between too close but very good fidelity and too distance with very different, with hallucinations everywhere? Okay, so this is a question, but it's not the only question, I would argue. So now I'm going to give you another visualization of a different experiment that we did. We worked then for a while with the 2017 Swedish population data and started to ask intersectional questions to that. So on the left here is a visualization of a question about the intersectional representation of highly educated women in Sweden. Where are they located? And you can see on the left that we have two purple spots. I'm sorry, this is a very small slide, but there are two purple spots on this graph. One is in the city of Uppsala, and one is in the city of Lund. And these are the two oldest university towns in Sweden. Definitely makes sense that you would have high densities of highly educated women in these towns. But then we synthesized the data and we just put it through I put it into the same visualization tool and asked the same question. Where in Sweden do you have high densities of highly educated women? And in that data set, those women appeared on the west coast of Sweden. Now, there are lots of women who live on the west coast of Sweden, and there are lots of highly educated people who live on the west coast of Sweden. There are also highly educated women who live on the west coast of Sweden for real, but the highest densities of them are not on the west coast of Sweden. So this is a plausible variation because it's completely plausible that you could have, and there are women with high educations in Sweden on the west coast, but it's maybe more than a simple plausible variation. I would argue that suddenly what we're seeing is a different ontological representation in that data set. Using the word ontology from more the philosophical way of like the thing that exists back beyond your data, beyond your methods of gathering it, that is then flattened and presented into data when you do gather your data. We can have that conversation in the chat afterwards if you want to, but like, I wonder if we need to start thinking about the representations in our synthetic data as more than simple plausible or implausible statistical variations of the original data, and rather new ontologies or multiple ontologies that we're producing in the production of synthetic tabular data. And if we do that, what does that do? Well, here's my argument. What it does is that I think it demands some guardrails. And I think those guardrails might be good, like might be well served by considering what sort of existing guardrails we have for data, like the FAIR principles, that sort of try to approach things like transparency and reproducibility. So when we were doing all this work, I was sitting there thinking, I wonder how data repositories are dealing with synthetic data sets and their hallucinations in them. Whether we want to call them intersectional hallucinations, or if we want to call them plausible and implausible variations, or if we want to go harder and say that we have new multiple ontologies in our data sets, I wonder how these data repositories are dealing with it. So I sent out a survey to a whole bunch of data repositories, most of them in Europe, some of them in the US as well, and just asked them, what kind of metadata or README requirements do you have for people who are depositing synthetic data sets with you? And the answer I got across the board was this. So I was like, okay, maybe we need to have a conversation about this, since obviously we are not having a conversation about this yet. And I understand because it's new and it's like probably not the biggest like problem with AI that we've got. And also, it's not the biggest problem for data repositories. This is just something small. And a lot of them answered back and was like, I don't think we have synthetic data sets. But I was like, well, actually, I sent this survey to you because I saw them in your data. But okay, fine. Let's just have a conversation. And the Swedish National Data Service was really open to that conversation, as was part of the UK Data Service. So we managed to pull together a series of workshops and seminars with people who work on metadata and READMEs, and then also people working with their machine learning teams at these data repositories, and just like having a conversation about what we think should be in the READMEs and the metadata for synthetic data sets. And we ended up publishing a couple of guidelines. The Swedish National Data Service has some recommendations. There's the AI Policy Lab published our guidelines with it. I have a spin-off because we're supposed to have spin-offs in Sweden. So I have this impact spin-off where we just put a programmer on the job and got them working on creating some sort of tool to make these things transparent and manageable for people generating synthetic data sets. But then we also use that spin-off to produce a README creator based on the workshops that we had with the data repositories, which is nothing complicated. It's just like you can QR code it with this little QR code and get to it. It's just basically like a form that ends up prompting people who are going to create a README for a synthetic data set that they want to deposit at a data repository to actually consider writing down some of the things that we think, given these workshops, they ought to have in their READMEs. And these are like I think there are things that are actually very relevant for regular data sets too, but definitely relevant for synthetic data sets, which means that most of them are based around the idea that you have to think about data as both a product and a process, right? So you have to think about the data in terms of like how it was gathered, what questions were used to gather it, what was the underlying hypothesis that directed the production of the data in the first place. How is it reusable and why? But then also, like with the synthetic data sets, what sort of models were used to generate it? For example, when we were using the GANs, we lost all of those edge cases. But when Sagi went over to using the diffusion model, she was able to control for the edge cases and keep them there. But it didn't happen automatically, even with the diffusion model, right? She still had to know which edge cases did we have in the data set, the original data set, and which ones did we want to maintain, and then make sure that the diffusion model was actually maintaining those edge cases. But then also, which intersectional hallucinations do we have? Like, will this data set be really relevant if you're asking questions about gender, age, and occupation? Or not? Or will this data set actually mislead you if you're interested in certain questions around like, your deep sea current, salination, and temperature? Like knowing the data before and after is really important. So putting that into this README or like some sort of metadata, I think is also really important. Anyways, if you get a chance, go out and check out the analyzer or the README creator, just because I would love to hear your feedback. This is an amazing audience. I would love to hear your feedback on what you think we are missing in it, what other things you think we should be including in a README for synthetic data sets, what else you think we don't need in there. And if you have any other ideas about things that we should be actually considering when we're thinking about synthetic data sets and sharing them. Though honestly, I'm also trying to get out there and think about like trying to express the idea that we need to think with synthetic data sets about, say, maybe an ABCD heuristic, that it should be accurate, or maybe more to the point, we should know how accurate it is and have that be clear, that it should be bespoke, that you can't just have one data set, synthetic data set, that then will be useful for everything. You have to know what your question is to that data set in the first place, and then create a synthetic data set that will answer that question while also allowing for the hallucinations or the plausible variations in it so that you can still call it a synthetic data set. And then it should be curated. You can't just like put it up on the cloud and then, boy, that was a boomer move, wasn't it? To like wave to the cloud, but put it up onto the cloud and then share it with everybody and just assume that it's going to be like, useful for everything. No. Curate it, know where it's being used, know what it's for, know how to, know how to package it, and then document it. Think about your data, your synthetic data, as both a product and a process, and document all of that and attach that to it so that you're doing something to making it fair, transparent, and reproducible. So anyways, again, like was mentioned earlier, this is not a single person job. I have a team that worked with me. I want to thank Francis Lee, Sagi Haji Sharif, Ying He, Richard Falk, Mons Gonzalez, and all of the funders, including the Wallenbergs. I know the Wallenbergs are funding things over there at Stanford, so they're doing a lot of that here in Sweden too. And thank you for giving me the chance to come and talk to you today and share some of our experiences with thinking about synthetic data and open science. Should I stop sharing so that we could open for questions?
You can, yes, definitely. We have a few questions in the Q&A. The first one is, all right, the first one is, at what point does abstraction degrade fidelity rather than enhance usability?
So, okay, I am the social scientist in the room. I wonder, how could you have something be usable if it didn't have fidelity? But you know why I ask this question? And I think this is really relevant for our conversations around synthetic data, but also data in general, is that when I think of data as a social scientist, somebody who's, somebody who's read all this work on like how the world becomes data and the politics of that, like, philosophical work about ontology and epistemology. So when I'm coming from that area, my whole take is that data is an incredibly interesting, powerful, and political representation of the world. And we have to know how it's representing the world for it to be useful, for us not to like do damage to the world by using it. But the approach, like the idea with data, is that it is and maintains fidelity at some level, and it will never be a good copy of the world. But it will be an attempt to produce a representation of the world. Now, if you're going to make a synthetic version of that, I really wonder what is the point of making a synthetic version of the world when you already have a half-good copy of it in the first place? But before my cynicism takes over, I will answer that question with, well, okay, Erica, but people using synthetic data, AI-generated synthetic data within this machine learning paradigm or rabbit hole that we're in, are using it not so much because they are interested in how it's representing the world, though maybe they should be, but not so much that, but because they're interested in how it's able to either blend with their real data or on its own work with the models. So from my perspective as a social scientist, and I think a lot of medical people too in the medical domain, their thoughts about data is that data represents the world, be that patients or diseases or something about the health of the world. Whereas a computer scientist is trained within a discipline and a domain that receives data, and that data is received as a fait accompli and as a packaged brick upon which to work and build their models. And they trust, they are not stupid, they are aware that it is still an incomplete representation of the world, but they trust that it is a good enough representation of the world to stop asking those questions and go forward and say, how can we manipulate this with whatever algorithms we have or whatever neural networks we're using or whatever large language models we want to use onto the data, and then see how it works with our models. So for them, the evaluation of the quality of the data is like a forward arrow. How is this useful? How can this be valuable for what we're going to do with it with our models? And that's a very different evaluative question or paradigm than the ones who are using the data as a representation of the world from which it was gathered. Now, I think that with the synthetic data conversation, we have an opportunity to say, maybe we need to pause and ask how we can conflate these two different bidirectional arrows into how we evaluate data and use them both in a conversation about what is important to gather in terms of creating synthetic data that could be useful and useful for both. Do I think there's an easy answer to that? No. But do I think there's ways to try to at least document the fact that we're asking this question? Yes. So that's why we've got this software that forces you to analyze your original data and be aware of things like the edge cases and the imbalances in it in the first place. And then through an iterative synthesizing process where you can choose different models and manipulate the types of models, what you're doing with the models, the heat and the weights and the training data and stuff, create new synthetic data sets that then you can easily compare visually to the original data sets. So you can see where your hallucinations are, see which types of edge cases have gone missing, and then that it's being documented in the background the entire time so that you can document this process as both a product and a process. It is the important thing for trying to engage a more robust conversation about synthetic data. That was a very long answer to a short question. I'm sorry.
It was a very complicated question, though. All right, next question. Can you comment on privacy measures that are relevant on a particular instance of a synthetic data set? For example, a differential privacy on the data versus generating the model itself?
No. And you know why? Because I have read a lot about this and I talk with people a lot about this and I've got a postdoc working on this, but honestly, no, because I think it's a terrifying thing to comment on. And the minute you start saying these words, you're going to have people who are saying, yeah, we can do that. And I'm just like, I don't know. I've heard that before. I don't know. I'm old enough at this point to think, yeah, there have been people promising solutions to these things for a long time. And there's always somebody else who's going to have an interest in getting around it.
Okay.
I'm sorry. Yeah, I'm
Okay.
sorry. Yeah.
Can you comment on using synthetic data to augment real-world data, especially with edge cases? Is this currently being done today?
Yes, it is currently being done today. And I want to applaud the desire to do that, not least because we all know that edge cases in our data are not always edge cases in the world. So trying to find a way to rebalance data sets that we know are not actually as representative of whatever it is that we're trying to measure and represent is a really, really good thing to try to do. So there's my thumbs up. But, and here's my gender studies professor coming in and saying, but it's probably much more complex than what you're thinking. So let's just be simple, and let's just be simple and think about like population data again. And let's be easy and just be like, okay, imagine we have a data set, and imagine we have a world that we've tried to represent in just a binary way, that we just have men and women, and we imagine that we have only 30% women in that data set, and we know that it should be 50%. And so we up the number of women to make it 50%. Those are our edge cases. We'll just up it up with synthetic data. But go back to those sploms I was showing you, right? And the way that as soon as you start creating synthetic data, you're going to be having intersectional hallucinations in that data set. You cannot get around it because you're shifting, even if you're like maintaining very good statistical representation in the intra-attribute columns, you're still going to be shifting how the inter-attributes, like the inter-attribute relationships are changing. And we all know that it's great to be, it's great to be a woman. Like, okay, whatever. It's great to have a data set that has enough women in it. But if you're just going to up that one column of women, are you also changing like all the actual really interesting elements, how being a woman in the world through the course of a lifetime in the power structures that we have change things like educational level or salary or marital status or like any of these elements. And especially then if you could add on to it, like say, for example, our ethnic background or our income level. How are you going to also up that in your data without merely reproducing, like copy and pasting particular data points? And copy and pasting data points is not what you want because then you're not actually adding more information to it. So I feel like it's a wonderful thing to want to use synthetic data to try to rebalance the known and discovered imbalances in your data set. I applaud that so much. And I know that this is impossible and expensive and painful, but maybe we also need to rethink how the original data was gathered in the first place and go back and try to address it through doing that. Oh, I feel like that cringy mom that just tells you to go and clean your room. Oh my God.
Along the same lines, could representational inequity become a public health or national security issue?
Representational inequity is a public health and national security issue. Partly because it reflects existing inequality, but largely because it represents epistemological inequality or epistemological violence. That is, that we do not see the categories of existence that we are not gathering data about. So by not seeing them, we're silencing them in our data there. But then, of course, which I assume is what you're actually getting at, by not having them in our data, we are, as we've discussed earlier this morning, training data, like training algorithms, training AI systems on them to imagine a world that is inequitably represented. So all of our AI data sets are being, AI data tools, all of our AI algorithms are being trained on data sets that are already displaying epistemological violence or silencing. And now we will be training tools that are oftentimes predictive models or whatever type of model that you're developing that are trained on a world that has already silenced these types of representation. And I think, and so I understand that we want to be able to use synthetic data to redress that, because we want to redress that because all of us want to do good with our work, right? And this is a great way to try to do it in theory, but I'm just concerned that maybe it's not actually going to be doing it well in practice. Or if it does, or maybe what I'm more concerned about is that we will think that we have done it well, and we will not have documented it properly, and we will share that with the world, but it will not have been done as well as we think it is, because actually inequality is so much more complex and reflects power dynamics in the world rather than mere data points.
Right. And here, the next question is longer, so I actually copied and pasted it into the chat box so you can see it. What a fascinating topic. Thanks, Dr. Johnson. I used to feed my casual inference algorithm synthetic data and real data and got completely opposite outputs. Upon reflection, I realized the possibility of bias in generation of synthetic data as well as collection of real data. And then I thought, is there a way to at least categorize the bias? Then, if they are different types, maybe mixing them two up can just offset the bias in each category. But does it make sense in your view?
I think it's so wonderful that you're trying. So this is what I want to say first. I think it's so wonderful that you care, that you're checking, and that you're trying, and please continue to do that. That said, bias, what do we mean by bias, right? Like go Google bias in machine learning or in any data science type of thing. We have so many different definitions of it. Many of those definitions counteract each other. So you would have to know what type of bias do you actually mean? How are you trying to address it? It's related to fairness. What kind of concept of fairness are you working on? These questions are just so incredibly complex. And I don't mean to say that to say stop doing it, but I do mean to say, boy, it's hard. Does it make any sense? It makes a lot of sense to keep trying. Definitely keep trying. We have to keep trying. It's not something I'm going to be able to answer in a Zoom meeting or probably even in real life. I think this is so hard, but so important. And here's the social scientist in me coming out as well. We have to stop thinking about bias and fairness as purely statistical or mathematical concepts with our data. And we have to take a page from a lot of the critical theory that we've been taught about how bias is a result of practice and a result of the ways that we are in the world and a result of the power dynamics that we live within. And we have to address that as well when we're trying to define what bias is. And in that sense, it's something that occurs continuously. We take a snapshot of it in our data, maybe at best, but it's a practice that is ensuing over time. And trying to put our fingers on that in our data is really hard.
Thank you. And we have two more questions. Is the future of human AI collaboration interface redesign or neural augmentation?
So I want to say interface redesign. But like I said, I've got this software that we've been developing, right? And every time I talk to somebody about using it, they're like, oh yeah, I want that to be automated. So I'm like, okay, great, we can do that. But then you're going to lose your ability to like try to know what's going on, right? What you're doing. So I don't know. Is it like maybe the future is a period of automation where it's just being automated, and then eventually we're going to have so much slop that people are like, oh, maybe we need to pause and look at what we've done and then try to put some human in the loop and an interface that works and then redo it. I don't know. Maybe it'll work. Here's the other, the social studies of science background that I have will actually remind me that we have done great science with bad data for ages. Like this is not new. So maybe we'll be able to do some good stuff too. I think we should know what we're doing. And that's the whole concept of fair data too, right? We should be able to know and explain to other people what we do.
All right, so then let's close out. I had two questions I was trying to decide which ones I should ask. Because this is a women in data science virtual event, What barriers are you still seeing for women in clinical and biomedical data science, and what concrete actions would make a biggest difference? I'm sorry, completely butchered it. Let me try it again. It's a lot of reading today. What barriers are you still seeing for women in clinical and biomedical data science, and what concrete actions have made the biggest difference in your own trajectory? Oh, I managed.
So I don't know if you can tell from my accent, but I am actually originally American. I grew up in Minnesota, and I moved to Sweden in my 20s and have been living here ever since. And for me, the biggest difference in my career trajectory has been that I moved to a country that had legally mandated gender equality, even if it hasn't been actually achieved, where it's something that we all agree as a collective that we want to try to do for the most part. And that has meant that structural things have been put in place, like very, very heavily subsidized childcare from the age of 1 and fully paid, fully shareable parental leave for up to 2 years, like 14 months to 2 years. So as a woman, I have been able to follow an academic career, get a PhD, go and do a postdoc, get a lectureship, work my ass off, publish like crazy, and teach. And also, since I'm just actually a really boring cis straight woman who's square and dull, find a husband, get married, and have a kid, and be able to do both of those things. Where I watch my friends back from the States who are trying to do the same thing, and it's much more difficult. So now I'm going to jump from being this I'm in the AI world talking about synthetic data, to just be like an old square 1980s structuralist sociologist and be like, God, you know what? We have to have systems in place around us that support all of us to be able to do the things we want to do with our life so that we can also do the things we want to do with our careers. That's probably not the answer you wanted. I'm sorry. But you have to be able to talk about these things with your friends and colleagues. And I'm not sure the environment you're living in at the moment allows that. So maybe try to work towards being able to use the words and terminology that are important for you to be able to make the changes in your life around you that you need. I'm sorry. Or come to Sweden. Move over here. We're very happy to have you.
As you can tell from my accent, I'm Hungarian, so I fully understand everything
You fully
just
understand.
said.
Yeah.
Yes. All right. Well, this takes us right at 12:05. Thank you so much for your presentation and for your insight. And thank all of our presenters today, our panelists and our opening keynote. Some of them are still on this call. I cannot express how grateful I am for your time and for sharing your expertise with us today. And there is a very quick, if you're inclined to do so, everybody will receive the, everybody will receive the recording and the slides within 48 hours. You will also receive the link to the feedback, to the conference feedback, or you can scan the QR code. It is anonymous. It takes less than 2 minutes to complete it. So if you could please do that, that will help us continue this event and making it better and better every single year. Again, I would like to thank my entire team here at Lane Medical Library. Without whom this event would never be possible, because they put in a lot of work. And just thank you. I really feel inspired after hearing your journeys and research efforts. And I, again, greatly appreciate the time and the effort today. So thank you so much. Thank you for being here. Thank you for asking questions from the audience, and hopefully we'll see you in future events. Thank you.